All right. Yeah, my computer restarted. So everything kind of everything's gone. Let's put everything in one tab then. That's okay. It is what it is. No, I don't know what are you doing. Again. I need to go back to the hub. Where's it at? This one. Yeah, nice. Okay, cool. All right, let's continue. We have the skin left. Maybe I think I'll do the flashcard for time. Wait, what? Over here. And yeah, I think that'll be that.
Cardiovascular system. In this next set of videos, we're going to talk about something called the integumentary system. The integumentary system. And what does that mean? I mean, I can understand what the cardiovascular system is, or the pulmonic system, or the renal system, but what is your integument? What comprises the integument?
Let me get some water before we officially start. Seriously. Not this one. Hold up. Which one though? Yeah, it's kind of a lot. I think it was this one. Yeah, this one. Cool. Alright, there we go. And, yeah. Yeah. These are these, right? Okay. Alright, we're good. Cool. Hey. Dope.
Integumentary system. And there are actually two things we talk about. The integumentary system is comprised of your skin as well as your appendages. Now, appendage, what does that sound like to you? An appendage could be something like something that hangs off or something that's a part of, like your arm is a part of your torso. Well, the appendages of your integumentary system involve things like your nails on your fingers and on your toes, your hair on your arm or elsewhere, and also things like your sweat glands or glands in general kind of fall under this classification. And we'll talk in detail about these appendages later, but I want to focus on the skin right now and do a bit of an overview. Because whether you recognize it or not, the skin is actually the largest organ in or on your body. It's 21 pounds. That's far heavier than your liver or your lungs. And yet many of us can go an entire day without thinking about the functions of our skin. Well, what are the functions of our skin? If I told you this was your arm right here, and this is you giving a little thumbs up, and then your fist, and then this going back here, what is it that the skin on your arm enables you to do? Well, one thing you may have noticed is that when it's raining outside, and you've got raindrops dropping on your head, one thing your skin enables you to do is be impermeable to the water. It's impermeable to water and other things that try and breach its layers and go into your organs or your bloodstream. It's impermeable. It cannot be passed. And that's as true for water and other molecules like this virus right here, which I'm drawing. Here's this little capsid, and its little legs wants to come in and infect the cells of your body. But thankfully, your skin says no to this virus, and it's not allowed to enter or breach this barrier because it's impermeable. But it's not just a structural barrier. It also has an immunologic function as well. Your skin can secrete things like antibodies or even enzymes like lysozymes. These are the guys that'll go and take on these viruses, or say if there's a bacterium that's present that has intentions of also getting you sick. The antibodies can coat this sucker right here, and the lysozyme can assist in breaking down the cell wall to help protect your skin. And so your skin also functions as part of the immune system. So immunity is also in play here, not just with this antibody or this lysozyme. Perhaps you can imagine a scenario where the bacteria maybe even penetrates a few layers of your skin and thinks that it has an opportunity to set up shop or make an infection or an abscess somewhere, but you have cells that are within your top layers of skin, like your Langerhans cells, like we'll talk about, that'll eat these bacteria up and prevent them from setting up shop or making you sicker than you should be. Other than immunity, your skin can also perceive things in the environment. If I have this little pin from a pin cushion that I can prick right here, and it really hurts right there, your skin will tell your brain, hey, maybe we shouldn't put our hand so close to this sharp object. So your skin also has the responsibility of perceiving your environment. And so it conducts sensation. It's able to tell when there's a stimulus that's either painful, so it perceives pain. It can tell different temperatures, whether something is hot or it's cold. And also it can differentiate types of touch that are present and discriminate between textures, whether something is just grazing your hand or something is poking you deeply. And finally, when you're outside and it's really hot, just kind of as I was already alluding to with the ability to perceive temperature, your skin has the ability to respond in a process that's called sweating, as we know it. We sweat because of our skin and the glands in our skin. And it's all part of an overall process known as thermoregulation. Thermoregulation, because our sweat allows us to cool off by a process known as evaporative cooling, but there are also processes that involve our blood vessels. Here's a blood vessel in your arm right here that can help us conduct heat out of the body. Let's get rid of that heat because we notice it's already hot outside. So the skin is so much more than just a barrier. There's a lot that it does for us. And in the next few videos, we'll go into detail about how all of these functions are achieved. They say that beauty is only skin deep. Did you know that the outermost layer of skin is actually composed of entirely dead cells? I have no idea what's beautiful about dead tissue, but in this video, we'll seek to figure that out. We'll talk about the different layers of skin. As you can see here, there are many layers to the skin, and there are 3 different sets of layers that we can talk about. There's the epidermis, which is the first 5 layers, the dermis, which is the next 2 layers, and then the last part, the subcutaneous tissue, or the hypodermis. In this video, we'll start by talking about the most superficial part of your skin, and that is the epidermis. And I'm sure your friends have told you before that your epidermis is showing. The epidermis is the topmost layer of skin, and itself is comprised of 5 layers, or as we call them, strata. So 5 layers or strata, and each strata or stratum has its own important characteristics. So we'll start from the bottom and talk about the deepest or bottommost layer of the epidermis, and that's known as the stratum basale, or the basal layer. The stratum basale sits right above the dermis and is the place where we generate what are called keratinocytes. And so I'm drawing a whole bunch of keratinocytes right here. These are keratinocytes. And in fact, the stratum basale has multiple layers of keratinocytes. And as you might recall, cyte just means cell. So what does keratino mean? Well, it comes from a protein or an intermediate filament that's called cytokeratin. Cytokeratin, which is something we first start making in the stratum basale. Sometimes it's just called keratin, and it helps give our skin its tough outer layer when we get to the top, as we'll talk about in a minute. And in other animals, keratin actually is the main ingredient for things like horns, or even hooves. So it's definitely the tough stuff that help protect our skin. And another very important thing to know about the stratum basale is this is where we have very rapid cell division. Rapid cell division because this is the bottom layer of our epidermis, so cells are made here and slowly they move upwards. Another thing that's important about the stratum basale is this is where we get our skin color from. So there are components of the stratum basale that determine what our skin color is going to be. And the cells that confer skin color for us sit here as well. And these guys, you may have heard about them before, are called melanocytes. Melanocytes. So melanocytes, and as you know, cytes again just means cell, but melano just indicates that there's a special pigment that's made in these cells that helps determine our skin color. And that pigment, as you may have guessed, is called melanin. Melanin. And the interesting thing about these cells or this pigment is that it's not the number of melanocytes that determine how dark your skin is. In fact, people with dark skin color and light skin color have the same number of melanocytes. It's actually melanin, the amount of melanin that you have, that determines how dark your skin color is, meaning that darker skinned people have more melanin. All right, so as we move up to our next layer over here, we see that our keratinocytes make it up here too, and we might start to notice some of this cytokeratin that's being produced here a little bit. And one of the unique features about this next layer is that we have this little connection that's starting to be generated in between our keratinocytes. This guy is called a desmosome, and this is involved in what gives this layer its name. So this layer that sits above the stratum basale is called the stratum spinosum. The stratum spinosum, which, as you might guess from the fact that we call this our basal layer, this stratum is our spiny layer. And the reason why we call it that is because of the desmosome. When we look at this layer of skin, sometimes underneath some microscopes where we have lost moisture or the water that is in our cells, we'll have these shrunken cells that actually look like stars or kind of like spiny cells, very pointy shapes. And the reason why they're pointy is because they're still connected by these desmosomes, but they've lost the water. So that's why sometimes these layers look shriveled. So shriveled here, and slowly they move upwards. That's known as the stratum basale, or the basal layer. The stratum basale sits right above the dermis and is the place where we first generate what are called keratinocytes. And so I'm drawing a whole bunch of keratinocytes right here. These are keratinocytes. And in fact, the stratum basale has multiple layers of keratinocytes. And as you might recall, cyte just means cell. So what does keratino mean? Well, it comes from a protein or an intermediate filament that's called cytokeratin. Cytokeratin, which is something we first start making in the stratum basale. Sometimes it's just called keratin, and it helps give our skin its tough outer layer when we get to the top, as we'll talk about in a minute. And in other animals, keratin actually is the main ingredient for things like horns, or even hooves. So it's definitely the tough stuff that help protect our skin. And another very important thing to know about the stratum basale is this is where we have very rapid cell division. Rapid cell division because this is the bottom layer of our epidermis. So cells are made here and slowly they move upwards. Another thing that's important about the stratum basale is this is where we get our skin color from. So there are components of the stratum basale that determine what our skin color is going to be. And the cells that confer skin color for us sit here as well. And these guys, you may have heard about them before, are called melanocytes. Melanocytes. And as you know, cytes, again, just means cell. But melano just indicates that there's a special pigment that's made in these cells that helps determine our skin color. And that pigment, as you may have guessed, is called melanin. Melanin. And the interesting thing about these cells or this pigment is that it's not the number of melanocytes that determine how dark your skin is. In fact, people with dark skin color and light skin color have the same number of melanocytes. It's actually melanin, the amount of melanin that you have, that determines how dark your skin color is, meaning that darker skinned people have more melanin. All right, so as we move up to our next layer over here, we see that our keratinocytes make it up here too, and we might start to notice some of this cytokeratin that's being produced here a little bit. And one of the unique features about this next layer is that we have this little connection that's starting to be generated in between our keratinocytes. This guy is called a desmosome, and this is involved in what gives this layer its name. So this layer that sits above the stratum basale is called the stratum spinosum. The stratum spinosum, which, as you might guess from the fact that we call this our basal layer, this stratum is our spiny layer. And the reason why we call it that is because of the desmosome. When we look at this layer of skin, sometimes underneath some microscopes where we have lost the moisture or the water that is in our cells, we'll have these shrunken cells that actually look like stars or kind of like spiny cells, very pointy shapes. And the reason why they're pointy is because they're still connected by these desmosomes, but they've lost the water. So that's why sometimes these layers look shriveled. So shriveled because they've lost the water, as well as spiny. And so that's how we remember the name, the stratum spinosum. Another type of cell that sits in this layer is an immune cell that is looking for foreign bodies or pathogens or things that can be destructive to our body, like bacteria and fungi, and eat them. These are called Langerhans cells. Langerhans cells. And as you may have guessed, these are part of our immune system. So they hang out here, but can definitely travel to other layers. We talk about them more in our dedicated immune system tutorial. All right, and so it's onward and upward to our next layer that's here. This more superficial layer is called the stratum granulosum. The stratum granulosum, which just means the granular layer. And the reason why we call it this is because the keratinocytes that sit here, now in addition to the desmosomes that we talked about from the spiny layer, have a tremendous amount of granules that are being produced here. And these granules are very apparent when we look at them under a microscope. These granules are called keratohyalin granules. So keratin hyalin granules. And I know what you might be thinking. So does this mean we're making a whole bunch more keratin in this layer? Well, we are, but these granules don't actually house keratin or the cytokeratin we talked about. They hold a whole bunch of other proteins that help handle our keratin. Just know that these granules make proteins that are best remembered as your keratin handling proteins. So your keratohyalin granules make your keratin handling proteins because they move your cytokeratin around in the cell. The other thing the stratum granulosum does is that it releases these things that are called lamellar bodies. And these lamellar bodies contain a whole bunch of lipids that then come together and form a strong lipid layer that sits at the top of our skin. This lipid layer is impermeable. It gives the skin its watertight capabilities that prevent foreign pathogens from making their way down deeper into our skin and eventually into our bloodstream. And so from there, we go to our next layer up to the stratum lucidum. The stratum lucidum, sure enough, this is our lucid layer, or our clear. I'll write that here. This is our clear layer. And the way I think about it was that in the stratum granulosum, our keratinocytes worked so hard to make these keratohyalin granules and release these lamellar bodies that they actually died by the time they make it up to the stratum lucidum. So these cells that we have here, these keratinocytes, are actually dead. These are zombie keratinocytes. And they look that way on microscope because they've lost their nuclei or other organelles that usually gave them the color we saw at our earlier, more alive layers. And so as a result, these layers will not have these granules or nuclei or organelles that we see in the previous layers. Instead, they're going to be far clearer or more see-through than what we saw earlier. And that's an important note to make off to the side. The stratum granulosum has lost their nuclei and organelles, which are usually supposed to give our cells color when we look at them under a microscope. And this trend continues when we move on to our topmost layer here. This is the stratum corneum. The stratum corneum, our topmost layer of the epidermis of our skin entirely. And the way I think about this is that this is the coroner's layer, the coroner. And as you know, the coroner's office is where dead bodies go for autopsies. And so this is also a layer of dead skin. And one of the key characteristics of the stratum corneum is that we'll have stacked layers of our keratinocytes, our dead keratinocytes. And we can have up to 15 to 20 stacked layers of these flat, simple squamous epithelial cells. So I'll write that off over here. We can have about 15 to 20 layers of our keratinocytes. Again, they're still dead because they died in the stratum lucidum. And because this is our topmost layer, we'll see that these cells will randomly as well as continuously slough off. So they will randomly and continuously slough off or leave, they'll fall off. And that makes way for newer cells underneath, or newer keratinocytes to make their way up to the top. Another interesting thing about this layer is that reptiles, I'll write this in like a reptile green. So reptiles will molt this layer or shed this layer off in one piece. And so that's why you'll see like snake skin that comes off as a single intact covering to the snake. So they molt off the entire stratum corneum, which is very interesting and also very gross. All right, so those are all your layers of your epidermis, all 5 of them, the top 2 being dead, and these bottom 3 or deeper 3 layers of the epidermis being alive. And as we move deeper down and start to talk about the dermis, those cells are also alive and have vessels in them to nourish them. We don't have vessels anywhere in the epidermis because our nutrients and our oxygen come from below and make their way upwards. We've talked about the epidermis, or our topmost layer of skin. Let's descend further down and talk about the dermis, these next 2 layers of skin. And then we'll also talk about the hypodermis, which is the bottommost layer of skin here. So as you can see, your dermis has 2 layers to it. And again, it sits right below your epidermis. So your epidermis, the topmost layer of skin, sits up here. And if you can recall, the bottommost layer of your epidermis is called the stratum basale, the basal layer of your epidermis. So below that, we have our first layer of the dermis, which is called the papillary dermis. This is the topmost layer of the dermis. And then below the papillary dermis, we have what's called the reticular dermis. The reticular dermis, right here. And the main difference between the dermis versus the epidermis is the type of tissue we have here. Recall that in the epidermis, we had epithelial tissue. So right, epithelial tissue up here. And then within the dermis, both layers are going to have something that's called connective tissue. You've probably seen that written out as CT, connective tissue. So connective tissue is different in that it doesn't necessarily involve cells. Epithelial tissue involves things like simple squamous epithelial cells, columnar epithelial cells, even transitional epithelial cells that you may have seen in the bladder. And your connective tissue contains a litany of other proteins, things like actin, which is what you must have heard of when we talked about the muscular system, or collagen, which is another structural protein. These are some of the most popular proteins you see mentioned when we talk about connective tissue, but they also include things like laminin, elastin, desmin. Connective tissue mainly holds things together. If you remember a tendon is connective tissue that holds the muscle onto a bone. And between the papillary dermis and the reticular dermis, there are two types of connective tissue we see here. In the papillary dermis, we have very thin, loose connective tissue. And this allows for all the stuff in the papillary dermis to sort of move around and change shape and position, versus what we have in the reticular dermis, which is thicker, more dense, or denser connective tissue, denser connective tissue. And the purpose of this is to make things stay still. We anchor things down in the reticular dermis. And we'll talk exactly about what we're anchoring down here in a moment. But as we hop back up to the papillary dermis, there are a variety of things that we have here that have to change shape and position and are able to do so because the connective tissue is so loose. One of the main things we find in the papillary dermis are blood vessels. You recall when you have an arteriole from an artery branch off into a capillary. So you've got these purple capillaries right here, and then come back together to make a venule, which goes off to a vein. And realize now that this is the most superficial or the topmost layer of your skin that actually has blood vessels here. So one of the roles is that the capillaries have to deliver oxygen and nutrients that nourish not only your dermis, but actually go up into the epidermis as well. Because the epidermis doesn't have any blood vessels. It relies entirely on the papillary dermis to nourish it. And thank goodness this is thin, loose connective tissue because there's plenty of room here for the capillaries to engorge with blood, fill up with nutrients, and then these nutrients can diffuse across and go up from the dermis into the epidermis and even go lower if necessary. The other thing we have in our dermis are nerve endings. These nerve endings, I'll write this just because my drawing's not as obvious, these nerve endings that sit in our dermis allow us to perceive things like touch and even pain. And then that will pass on a signal across the nerve going in this direction right here, like that. And this signal is relayed all the way to a direction right here, like that. And this signal is relayed all the way to a cell body. And this cell body can sit somewhere much further away than the skin. Now, as we get to the reticular dermis, we'll see why it's important to have thick, dense connective tissue down here. And this connective tissue helps anchor things down. One of the things that you can have, and I'll sort of draw it like this, ascending to your epidermis, and so it leaks out over here. This is a gland. You'll have tons of sweat, glands and exocrine glands of other varieties that start in your reticular dermis and are anchored and held down here that extend out with a duct to secrete their contents to the epidermis, to the outside world, kind of at the top layer of your skin. So you've got plenty of glands that sit here. In addition, you'll also have this follicle right here. This follicle will have from it emerging a hair. This hair, I guess I should label this also. The hair, of course, is going to protrude at the top of your skin. It's not that you just have a hair that sits inside. And then this guy right here is just your hair follicle. And so your hair follicle is anchored within your reticular dermis, and the hair itself is allowed to grow and extend out to the top and protrude from your epidermis. One of the other things that we have kind of sitting in your dermis that's anchoring itself onto the hair or the hair follicle is this slab of muscle that we've got right here. So this guy, I'll write it off there. This is an erector pili muscle. An erector pili muscle, and this is involved in extending or having your hair stand straight up when you're cold or when you're scared. And finally, now that we've talked about the dermis, we can move to our last layer of skin. Although it's technically not skin, but it's called the hypodermis. This is the hypodermis, which is a great name because, as you might recall, hypo just means below. This layer sits right below the dermis. Another name for it is subcutaneous fat, and I think that's also a great name for what we have here, subcutaneous. If you might remember, cutaneous is just another name for skin, so subcutaneous fat. And that's exactly what we have down here. We have tons of layers of fat that could vary from person to person. And although fat gets a lot of flack, we have to realize that it's very important because there are two main functions they help us achieve. First of all, fat absorbs shock. People that are more insulated with fat are able to withstand traumatic injuries maybe more effectively than others. And the other thing that it does for us is that it insulates our tissue. Because realize that now that we've finished talking about our skin, we've talked about the epidermis, the dermis, and now we've even covered the hypodermis. What sits right below this? You're going to find muscle. Muscle sits down here. It's below that, and perhaps even bone. You know, if you're talking about your chest wall right above your rib, if you put your finger right kind of where your clavicle is above your rib cage or even on your ribs, realize that your epidermis, dermis, and your hypodermis are all that separate your finger from your rib bone, or even your muscle if you're pressing on your bicep or your calf. So we spend a lot of time kind of talking about the components of what's in our skin, but how does this even help us clinically? How do doctors use this to diagnose diseases? The best one to talk about as an example here would be for the diagnosis of burns. Now, a burn is a horrible thing that could happen, and one of the things that we do to differentiate the types of burns you can have is to say what degree of a burn you've sustained. And interestingly enough, the different layers of your skin correspond to the different degrees of burn you can have. You can have a first-degree burn, which is just sort of a reddening that can occur on your epidermis. You can have a second-degree burn, which is characterized by burning into the dermis, so much so that you don't have nerve pain after some time. That's the difference between a first degree and a second degree. First degree hurts awfully, but if you have a place that's been burned that doesn't hurt so much anymore, it's because you've burned and killed off nerve tissue here, and so they're not able to send a signal up to the cell body. And then finally, there's a third-degree burn, which is going past the hypodermis into things like your fat, the muscle that's below, the bone that's below as well. And of course, the third-degree burn is characterized by not having any pain and having more of a darker coloration to it, because now you're burning more than just connective tissue. You're actually burning bone or muscle or fat as well. And it's this exact anatomy right here that we've talked about that allowed doctors to distinguish between whether a patient has sustained a first, second, or third-degree burn. And that's something you can do now as well. So to build on our knowledge of the integumentary system, we understand that integument is made up of layers of our skin, as well as things that are called appendages. I'll write that up here.
Hey, Hassan. Much love, brother. Much love.
Appendages. And appendages is kind of a loaded term. There are a lot of things that fall under this classification. Your nail, for instance, is an appendage. So how does your nail even grow? Well, why don't we just blow up this interface right there to get a better sense of how our nail is structured. So let me just draw your finger right here, and it's going along this way, and then I'm going to draw without the nail for right now, so you can sort of see how it grows, and this is kind of where your finger is coming off. The first thing to realize is that this part right here, which is called the nail root, is attached to your epidermis. So I'll color it blue because that's how we've been labeling our epidermis so far right here. And I'll even write it out. So this is your epidermis. It's your topmost layer of skin that we talked about in a separate video. And this is attached right to your nail root right there. So that's your nail root. And the interesting thing about this interaction point right here is that when you have cells grow in your epidermis, remember that you have cells ascend from your deeper layers up to the top. But here, where the nail root is, you'll also have cells that grow out that way. So you're going to have cells that come from the stratum basale, if you remember that term, for the bottommost layer of your epidermis. And you'll have some of the keratinocytes grow, die, and then eventually extend into the nail. And what that means is that your nail is essentially part of the epidermis. So I'll draw your nail right there in blue because it's made up of thick keratin. So keratin from your keratinocytes are the cells that made up your epidermis. And this keratin is just packed into a whole bunch of dead cells. So the dead cells that sit at the very top of your epidermis hold or are packed with keratin, actually. They're packed with keratin and they move to the top of the epidermis and then they kind of shift this way. And recall the keratin is the reason why your skin is so tough. And in fact, in the nail, the keratin is what keeps the nails so stiff. And this is true for your fingernails and your toenails. The difference is your fingernails actually grow about 4 times faster than your toenails. And I guess just for reference, because we're going to be talking about it in our other appendages, remember that below your epidermis, you're going to have your dermis here, and below the dermis, there's the hypodermis, or the subcutaneous fat or tissue. Great, so that's how our nail is structured and grows. Let's talk about another appendage that might be back here, for instance. So hair. Let's say we're talking about this back-handed hirsute right here, which just means somebody that's got a lot of hair on the back of their hand. We'll do the same trick and blow this up to get a better look at what's going on where our hair grows. So the big difference between our hair and our nails is that our hair grows from the dermis. So I'll draw our skin right here, sort of the same orientation we had down here. And recall that our epidermis is our topmost layer of skin. I'll make it small here because it's not going to be most of the business we're talking about. And then below it, we've got our dermis right here. And I'll label that because it's important. That's our dermis. This is where we're going to be talking about most of our stuff right now. And then below the dermis, remember, there's our subcutaneous fat, or the hypodermis. And so the thing about the dermis to recall is that there are 2 main parts to it. We'll kind of just draw it like that. There's the papillary dermis. All right. Dermis papillary, just the top layer of the dermis. And then there's the reticular layer. If you recall, the papillary layer is the thinner, looser connective tissue layer. And the reticular layer is the thicker, more denser connective tissue layer. So the way our hair is set up is that there's a follicle that originates here. It's this bulb that sits in your reticular dermis. So I'll label that off here. This is your hair follicle. You may have heard of that term before. Your hair follicle sits in the reticular dermis. And then from this follicle, you've got this hair, we call it the shaft of your hair, that extends upward and out. And realize that just like the skin and with the nail, the hair itself has a whole bunch of flattened or stratified squamous epithelial cells that are filled with keratin. And so the keratin is inside of the cells that are stacked up here in the hair that I'm coloring in. And it also is surrounded by keratin. So there's your hair, and actually your hair grows at about a rate of 0.5 inches or 1.25 centimeters a month. So let me label this right here. This is our hair shaft that you can see protruding through your reticular dermis into the papillary dermis, into the epidermis, and even to the external environment right here. The other thing I need to draw now, aside from the hair follicle and the shaft, is this band of muscle that I'm going to be putting in right here. It's important that I mention that it sits here in the papillary dermis. So it's in this top layer of the dermis right here. This band of muscle, right all the way here, is called the erector, with an A, erector pili muscle. Erector pili muscle. And if you'll recall your types of muscle, this is smooth muscle. This is not under our control. We can't cause our erector pili muscle to contract. Something happens involuntarily. And when this muscle contracts, there are 2 things that happen. One, you'll have your skin sort of bunch up together, and so you'll have what's commonly referred to as goosebumps. And you can see that here, because if this band of muscle gets shorter or skinnier, you'll just bunch your skin together and you'll have little lumps that come up here. So you'll have a lump that shows up, and that's your goosebump. And the other thing that'll happen is that it'll make your hair stand up. It'll make your hair stand up. So I'll just write hair stand. I don't have as much space here. And your hair will stand up. And this occurs either from strong emotion or exposure to a cold environment. So one thing you might be asking is, why do we even have this erector pili muscle? What's the point of having our hair stand on end? And actually, this is something that's more useful in animals than it is for us. So imagine if we're talking about a polar bear. I'll give it my best shot at drawing a paw right here to represent a little polar bear. But we've got this polar bear right here, and this guy's in the cold. So poor guy's out in the cold, okay? And when a cold breeze hits this guy's little paw right here, what's going to happen is that the hair on this polar bear is going to stand on end like that, just like I talked about in the case of a human being. Now, the hair in a polar bear is a lot longer than the hair in a human being. And by making this furry coat stand up like this, what happens is you create a warm, insulating layer. The layer of air that's embedded within the hair right here is significantly warmer than the external cold environment. And so what that does is that allows the warmth to stay within the paw and also extend to the rest of the polar bear's body, so the polar bear stays warm. We don't have that much hair. And so the erector pili muscle, to some extent, is actually a vestigial structure. Vestigial meaning currently useless or not functional anymore. But it's still a pretty interesting reminder of what other animals would do to deal with the cold and how we sort of try to manage without their abundance of hair. So to continue our discussion of appendages, I'll come to this palm side right here. And so I'll draw this little water droplet to indicate this person has a very sweaty palm. And the reason why it's so sweaty is because this guy has a lot of sweat glands. So a lot of sweat glands that are on his palm or maybe elsewhere in his body. And as I mentioned in an earlier video, our glands are situated in the reticular dermis. They're in the reticular dermis, and they have ducts that lead out into the external environment to help coat our skin with their secretions. And now we have three types of glands that exist in our skin. They're called the holocrine gland, there's the apocrine gland, and finally the merocrine gland. So I'll kind of compare and contrast them here. The first thing to realize is that the way we name these types of glands indicate how they release their secretions. The holocrine gland releases its secretions by disintegrating the whole cell. So the entire cell breaks down to release what's called sebum. Sebum. And so another name for holocrine glands are sebaceous glands. Sebaceous because they release sebum. Now, apocrine glands release secretions from the apex of the cell, or the top of the cell breaks off. So the top of your cell breaks off to release a mixture of proteins, lipids, and steroids. So proteins, lipids, and steroids. So a whole bunch of things right here. And finally, merocrine secretion or merocrine glands release their secretions, which is mostly the watery sweat we think of. These are the main sweat glands we talk about. They release watery sweat by a process you may be familiar with. It's called exocytosis. That's the budding off of the cell membrane with contents from inside the cell, which in this case would just be watery, salty contents that would be sweat. Now let's get a little more room right here and talk about where each of these types of sweat glands are found. Holocrine sweat glands are found on the face, the chest, and the back. The face, the chest, and the back. It looks like I didn't write it here, but I want to make sure you know that sebum is mostly an oily substance, so it's more lipid concentrated than, say, something like your apocrine secretion, so it's very oily. And now your apocrine glands are mostly concentrated in your armpits, the groin, and around the nipples. The armpits, the groin, and around the nipples. And the main thing here that's unique about the apocrine sweat glands is that they release their contents into the hair follicle. So they release their contents into the hair follicle, which is unique from the other types of sweat glands we're talking about. And then finally, our merocrine glands, which are our bread and butter sweat glands, are found literally everywhere else in the body, and they're mostly concentrated in our palms, our hands, like this guy up here, palms and our hands and even the soles of our feet, our palms and our soles. And then finally, what are the functions of these three types of glands? Well, for our holocrine or our sebaceous glands, this oily or lipid-rich sebum is meant to lubricate the skin, so it lubricates our skin. And in doing so, that also slows bacterial growth. So it slows bacterial growth by making it difficult for bacteria to move around and divide. So it slows bacteria growth. Now for apocrine glands, the interesting thing about them is that they don't even really begin to release secretions until after puberty hits. Until after puberty. And the reason why could be better understood when we talk about animals, because apocrine glands in animals release things like pheromones and warning signals and even territorial marking signs that we don't really practice as human beings. So the interesting thing about apocrine glands is that they're involved in what's called emotional sweating. Emotional sweating. They're an ongoing topic of research. Emotional sweating, meaning that these glands are most active during times of great anxiety, stress, fear, and even pain. And finally, our merocrine glands are actually, I'd say, our most important glands because they help us cool down through evaporative cooling. We talk about that in a separate video. So they help us cool down with our sweat. They help us eliminate waste products as well. So any extra nitrogenous waste products that we might have, or water or electrolytes we don't need, we release through our merocrine glands in our sweat. And finally, merocrine glands also release things like lysozymes, which are enzymes that will lyse bacteria, and even antibodies, which are immunologic proteins, so antibodies that'll help tag bacteria and other foreign pathogens or bad stuff that could cause damage to our skin and our bodies, tag these things for elimination. So your sweat is far more important than just cooling you down. It even helps your livelihood, in addition to all the other things we talked about here. So there's a lot going on when we talk about our glands and the rest of the appendages we've appreciated here.
Why does sweating cool you down?
Why does sweating cool you down? That is an excellent question, LeBron. And to answer it, let's zoom in on a droplet of sweat. And sweat is mostly water. So when we zoom in, we've really zoomed in even more than I've drawn over here. When we really zoom in, we'll start seeing mainly these water molecules. And the water molecules, just to be a little bit more accurate, I've drawn the oxygen in blue, and then the hydrogens that are bonded with that oxygen I've done in white. We all know that water is sometimes referred to as H2. That's for the two hydrogens, H2O. So each of these are a molecule of H2O or a molecule of water. What I've drawn down here, and this is an oversimplification of the molecules of your skin, but just for simplification, these are the molecules of your skin, really the parts of the skin cell. These aren't even the skin cells themselves. These are the molecules that make up the skin cells. And right over here, these are molecules of sweat, or it's really just molecules of water. So the question of why does sweat cool you down could really be restated as, why does having water on the surface of your skin actually cool you down? And to answer that, or to think about that question, we have to think about what it means to have temperature, or what temperature even really means. Temperature, what we perceive as temperature, is really just the motion of the molecules of something. So higher temperature means that they're moving around more. So high temperature, they're moving around more. Low temperature, they're moving around less. And they can move around in different ways. They can have what you could call translational motion, which is they're actually moving around. They could be vibrating. They could be rotating in some way. And in general, on average, the more of this motional energy, often referred to as kinetic energy, that these molecules have on average, the higher the perceived temperature would be. Now, how does having this water here cool down the skin? And well, first of all, why is the skin warming up? Because the muscles are doing all of this work. They're releasing heat. That heat is being transferred to the skin. But then how does having this water here help? Well, this skin has a certain temperature, a certain kinetic energy or motional energy. But when we say that, it doesn't mean that all of these molecules have the exact same motion. The temperature is the average motion. Some of these are bumping around at a faster speed, or vibrating at a faster speed, or rotating at a faster speed. Some are doing it at a slower speed. But as these bump around, they're going to bump into these water molecules and start to get them moving around. They will probably be moving around a little bit to begin with, but then the warmer this is, the more energy here, they'll bump into these molecules. So let's say that guy bumps into that, then he'll bump over there. And so that energy, this bumping energy or this kinetic energy, some of it will be transferred, or you could even say some of that temperature, some of that heat will be transferred to these water molecules. But the important thing to remember is this is a really kind of crazy thing. They're all bumping into each other and rotating in all sorts of crazy ways. They will have an average kinetic energy, which we perceive as temperature, but they all have, this one might be going really, really, really fast in that direction, while this one might be going really, 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 really slow. This one might be going really, really, really fast in that direction. This one might be going really slow in this direction. So the thing to think about is, Given that you have all of this variation in the energy of each of these particles, which of these are most likely to escape, to actually evaporate? And to think about evaporation, you just have to think about that most water molecules, or the water molecules that are in that droplet, they do have an attraction to each other. We call those hydrogen bonds. So they do have an attraction to each other, and that's why a droplet kind of sticks together. But if one of these molecules is moving fast enough, and if it's moving in the right direction, it has a higher probability of being able to escape, being able to actually escape that droplet. And this process of these molecules escaping, that's what we refer to as evaporation. Evaporation. If a molecule has enough energy, it will escape the bonds with the other water molecules, and just evaporate into the air. But we still haven't fully answered our question. So let's say that this one is one that has evaporated and has fully escaped. Why would that actually be able to actually escape that droplet? And this process of these molecules escaping, that's what we refer to as evaporation. Which of these are most likely to escape, to actually evaporate? And to think about evaporation, you just have to think about that most water crazy ways, they will have an average kinetic energy, which we perceive as temperature, but they all have, this one might be going really, really, really fast, while in that direction, while this one might be going really, 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 really slow. This one might be going really, really, really fast in that direction. This one might be going really slow in this direction. So the thing to think about is, given that you have all of this variation in the energy of each of these particles, which of these are most likely to escape, to actually evaporate? And to think about evaporation, you just have to think about that most water molecules, or the water molecules that are in that droplet, they do have an attraction to each other. We call those hydrogen bonds. So they do have an attraction to each other, and that's why a droplet kind of sticks together. But if one of these molecules is moving fast enough, and if it's moving in the right direction, it has a higher probability of being able to escape, being able to actually escape that droplet. And this process of these molecules escaping, that's what we refer to as evaporation. Evaporation. If a molecule has enough energy, it will escape the bonds with the other water molecules, and just evaporate into the air. But we still haven't fully answered our question. So let's say that this one is one that has evaporated and has fully escaped. Why would that actually cool down this entire system? Why would it cool down the droplet and essentially give it more capability to accept more energy from the skin? Well, we just said the ones that have the highest energy are the ones that are most likely to escape, the ones that have the highest kinetic energy. So if you have a bunch of stuff, some are fast, some are slow, vibrating a lot, vibrating less, but the ones that have a high kinetic energy are the ones most likely to escape. What happens when they escape? Well, then the average kinetic energy will go down, or you could say the temperature, which is really just the average amount of motion or kinetic energy that's in this droplet. If the really fast ones, the ones with a lot of energy, are leaving, then the ones that are left over are on average going to have a lower kinetic energy or a lower temperature. And so that is what is cooling you down at a molecular level. Let's first think.
Why do we shiver when it's cold? What do the 5 fingers say to the face? In this video, we'll talk about how our body uses our muscles to maintain a cold body temperature. That process is called thermoregulation, the regulation of body temperature. So in order to do that, I'll first give you guys an overview of how thermoregulation works. Next, we'll talk about how our bodies respond to hot temperatures before finally talking about how our bodies respond to cold temperatures. So first, if we have this gentleman right here who doesn't look very impressed, and he's holding his hand up one day and notices it's really hot outside. It looks really hot. So the skin on his hand and his arm, the rest of his body, will perceive that it's really warm. And in doing so, this will create a neuronal signal, a signal that's sent across neurons up into the brain. And the part of the brain that perceives that it's really warm outside has a specific name. It's called the hypothalamus. The hypothalamus. And in fact, we split up the hypothalamus into 2 different parts to respond to 2 different types of temperature. There's the anterior hypothalamus, which just means the front of the hypothalamus. And then there's also the posterior hypothalamus, and that's just the back of the hypothalamus. But which one responds to what kind of temperature? Well, the way I remember it is that if it's hot outside, we like to use the front of our hypothalamus, or the anterior part of our hypothalamus, to respond to temperatures that make it feel like we're on fire. So if we're on fire, we're going to use the front of our hypothalamus to respond. Well, what if it's cold? In those situations, we'll use the posterior part, or the back of our hypothalamus, to respond to climates that make us say, brr. We say brr because it's really cold outside, so we use the back of our hypothalamus. Okay, so that all sounds good, but what happens after our brain knows that it's really hot outside? Well, then it sends back a signal to our bodies telling it to respond to maintain our core body temperature. And of all things to take point from here and act, it's going to be muscles in our body that will do something to make sure we can maintain our core body temperature. That'll involve smooth muscle, and when it's cold, we also use skeletal muscle to respond to changing temperature. So how does that work? Let's take a look. When we're in a hot or a cold environment, how do muscle help us respond here? Well, the way to talk about this is to go through the two main types of muscle that'll act here. First, there's smooth muscle. Specifically, it's smooth muscle that lines our arterioles. Now, arterioles are just smaller versions of our arteries, arterioles. The other type of muscle that functions here is called skeletal muscle. And that's the type of muscle that works in your biceps, your triceps. All right, now let's answer this question here. Why do you think arterioles have anything to do with temperature? I mean, how does that change whether we have a lot of heat in our body or not? Well, the way that works is that if you take a look at a blood vessel, I'll just draw a small one right here. It's got a bunch of red blood cells and white blood cells and proteins and whatnot. All of this is just kind of running around through the bloodstream. And the way it does that is just kind of in a random sort of motion or manner. It's never directly forward, but it just kind of bumps around like that and moves this way. So overall, you've got a general movement in the forward direction, but you kind of are bumping around to do this. And the interesting thing is this movement here, this momentum that your red blood cells, white blood cells, or whatever you have in your bloodstream make, that's energy. So we have a ton of energy in our bloodstream. And energy is just another form of heat. So if we are in a hot environment, we want to get rid of this heat. So we're going to put more heat to our most superficial or the most external parts of our body, our skin. So the more blood flow we get to our skin, the more heat we can have leave our bodies. And that's how we get to regulate our temperature. So let's talk about it here. When it's hot, our smooth muscle then is going to relax. And in doing so, the arterioles then are going to become wider or larger. And this process is called vasodilation. Your arterioles are dilating. They're becoming wider. And so the way you could see that happen is that this blood vessel that was about yay big is now going to become that big in the skin. And so that means that you're going to have a lot more blood flow going in here to the skin. You're going to have tons of red and white blood vessels and protein just kind of merrily tumbling about going like this. That means you're going to have a ton more heat or energy that's present here that can then dissipate and leave. And so that helps you cool off. You're losing all of this energy or this heat that you have by diverting blood flow to your skin. Now, what does skeletal muscle do here? They don't do anything when it's hot. They'll respond in a second, and we'll talk about that when it gets cold. What do smooth muscle do when we're in the cold? Well, you can imagine that if we relaxed when it was hot, it's fair to think that we're going to contract when it's cold. And so this process then in the arterioles is going to be called vasoconstriction. Vasoconstriction, which just means the narrowing of our arterioles. And so if I were to draw that on a diagram right here, you would see that this blood vessel that looked like this in our normal state is now going to turn into something more like that, kind of a pipsqueak, really tiny. So you're going to have red blood cells, white blood cells, and protein just kind of shifting through here like this, but there's not a lot of opportunity to kind of tumble around and be naughty. They just got to go forward. They can only go in one direction because there's not a lot of space here for blood flow. And so in the skin, you're going to have less energy or less heat that's present at this very external or superficial part of your body. The more blood that you have that gets away from the skin, the more that's going to go towards your core. Your core will then have more energy or more heat to help you stay warm when it's cold outside. So that's what your smooth muscle does. What do skeletal muscle do? Well, skeletal muscle will also contract when it's cold. But there's a very different purpose for why that happens. When skeletal muscle contracts, it's going to take ATP, or adenosine tri or 3-phosphate, and break that to make adenosine diphosphate, ADP. So that's 2 phosphates. And we just snapped off a little phosphate group. I'll write an I here to show that it just snapped off. But these aren't the only 2 things that are made. You actually will produce energy as well. And we call this reaction an exothermic reaction. Exo meaning exiting or the leaving of, thermo or thermic meaning heat or energy. And so we're actually producing energy here by contracting our skeletal muscle. This happens in our core muscle groups that cause more of the heat that's being produced from this reaction to be stored there to help us respond to these cold environments. Now, this isn't such an unusual process that's so foreign to us. The contraction of our core skeletal muscle groups is actually just called shivering. This is the whole reason why we shiver when it's cold, because we need to produce some energy by breaking off some phosphate from this ATP, and that shivering then will help us warm up when we're in the cold. And that's how our muscles help us respond to temperature change.
Be back.
Hey, thanks, Bara. Yeah. I'm gonna be right back, guys. I'm gonna get some in and out. So let's see how long it takes me. I'm gonna do drive-through though, so we'll see. I'll be back. Maybe like 25 minutes or something. Let's see if I can be back by like 2:10. Be back. 2:10. What the heck? Where's the Took way longer than expected, eh? Yeah, at least we got the stuff. All right, let's see first, and then we'll get, and then we'll get started with the rest of it, huh? I mean, it's 3 lessons. It's what, 4 lessons? 1, 2, 3. It's 3 lessons, and then I want to do the questions. I think today I'll start reading the book, and then we'll do Yusuf and go from there. Probably chemistry next. Well, I've been saying chemistry next, and then psych and soc. Psych and soc. All right, break. Just break.
in my humble opinion, the single most important biochemical reaction, especially to us, is cellular respiration. And the reason why I feel so strongly about that is because this is how we derive energy from what we eat, or from our fuel, or if we want to be specific, from glucose. At the end of the day, most of what we eat, or these carbohydrates, end up as glucose. In future videos, I'll talk about how we derive energy from fats or proteins. But cellular respiration, let's go from glucose to energy and some other byproducts. And to be a little bit more specific about it, let me write the chemical reaction right here. So the chemical formula for glucose, you're going to have 6 carbons, 12 hydrogens, and 6 oxygens. So that's your glucose right there. So if you had 1 mole of glucose, right, that's your glucose right there. And then to that 1 mole of glucose, if you had 6 moles of molecular oxygen running around the cell, then, and this is kind of a gross simplification for cellular respiration, I think you're going to appreciate over the course of the next few videos that one can get as involved into this mechanism as possible. I think it's nice to get the big picture. But if you give me some glucose, if you have 1 mole of glucose and 6 moles of oxygen, through the process of cellular respiration, and so I'm just writing in kind of a big black box right now. Let me pick a nice color. So this is cellular respiration, which we'll see is quite involved, but I guess anything can be if you want to be particular enough about it. Through cellular respiration, we're going to produce 6 moles of carbon dioxide, 6 moles of water, and this is the key, this is the super important part, and we're going to produce energy. We're going to produce energy, and this is the energy that we're going to that can be used to do useful work, to heat our bodies, to provide electrical impulses in our brains, whatever energy, especially a human body needs, but it's not just humans, it's provided by this cellular respiration mechanism. And when you say energy, you might say, hey, Sal, in the last video, didn't you just, well, if that was the last video you watched, you probably saw that I said that ATP is the energy currency, for biological systems. And so you might say, hey, well, looks like glucose is the energy currency for biological systems. And to some degree, both answers would be correct, but to just see how it kind of fits together is that the process of cellular respiration, it does produce energy directly, but that energy is used to produce ATP. So if I were to break down this energy portion of cellular respiration right there, some of it would just be heat, it just warms up the cell, and then some of it is used, and this is what the textbooks will tell you. The textbooks will say it produces 38 ATPs that can be more readily used by cells to contract muscles or to generate nerve impulses or do whatever else, or grow or divide or whatever else the cell might need. So really, cellular respiration, to say it produces energy, a little disingenuous. It's really the process of taking glucose and producing ATPs with maybe heat as a byproduct, but it's probably nice to have that heat around. We need to be reasonably warm in order for our cells to operate correctly. So the whole point is really to go from 1 mole of glucose, and the textbooks will tell you, to 38 ATPs. And the reality is this is in kind of the ideal circumstances that you'll produce 38 ATPs. I was reading up about it a little bit before doing this video. And the reality is, depending on the efficiency of the cell in performing cellular respiration, it'll probably be more on the order of 29 to 30 ATPs, but there's huge variation here, and people are really still studying this idea. But this is all cellular respiration is. In the next few videos, we're going to break it down into its kind of constituent parts, and I'm going to introduce them to you right now, just so you kind of realize that these are parts of cellular respiration. The first stage is called glycolysis. Glycolysis, which literally means breaking up glucose. And just so you know, this is part, the gly for glucose, or the glyco for glucose, really. The glyco for glucose. And then lysis means to break up. When you saw hydrolysis, it means using water to break up a molecule. Glycolysis means we're going to be breaking up glucose. And in case you care about things like word origins, glucose comes from the gluc part of glucose comes from Greek for sweet, and glucose is indeed sweet. And then all sugars, we put this "-ose" ending, so that just means sugar. So you might think it's kind of a redundant statement to say sweet sugar, but there are some sugars that aren't sweet. For example, lactose. Milk, it might be a little bit, but when you actually digest lactose, then you can turn it into an actual sweet sugar, but it doesn't taste sweet like glucose or fructose or sucrose would taste. But anyway, that's an aside. But the first step of breaking up glucose, and in case you care about things like word origins, glucose comes from the gluc part of glucose comes from Greek for sweet, and glucose is indeed sweet. And then all sugars, we put this "-ose" ending, so that just means sugar. So you might think it's kind of a redundant statement to say sweet sugar, but there are some sugars that aren't sweet. For example, lactose. So, milk, it might be a little bit, but when you actually digest lactose, and you can turn it into an actual sweet sugar. But it doesn't taste sweet.
Gotcha.
Like glucose or fructose would actually, or sucrose would taste. But anyway, that's an aside. But the first step of cellular respiration is glycolysis, breaking up of glucose. And what it does is it breaks up the glucose from a six carbon molecule. So it literally takes it from a 6-carbon molecule. Let me draw it like this. A 6-carbon molecule that looks like this, and it's actually a cycle. Let me show you what glucose actually looks like. This is glucose right here, and notice you have 1, 2, 3, 4, 5, 6 carbons. I got this off of Wikipedia. Just look up glucose, and you can see this diagram if you want to kind of see the details. Where you can see I have 6 carbons, 6 oxygens. That's 1, 2, 3, 4, 5, 6, and then all of these little small blue things are my hydrogen. So that's what glucose actually looks like. But the process of glycolysis, you're essentially just taking I'm writing it out as a kind of a string, but you can imagine it as a chain, and it has oxygens and hydrogens added to it, to each of these carbons. But it has a carbon backbone, and it breaks that carbon backbone into 2. That's what glycolysis does, right there. So you kind of lysed the glucose and each of these things. And I haven't drawn all the other stuff that's added onto that. These things are all bonded to other things with oxygens and hydrogens, whatever. But each of these 3 carbon backbone molecules are called pyruvate. We'll go into a lot more detail on that. But by glycolysis, it by itself generates, well, it needs 2 ATPs. So it needs 2 ATPs, and it generates 4 ATPs. So net, on a net basis, it generates 2, let me write this in a different color. It generates 2 net ATPs. So that's the first stage. And this can occur completely in the absence of oxygen. I'll do a whole video on glycolysis in the future. Then these byproducts, they get re-engineered a little bit, and then they enter into what's called the Krebs cycle. They enter what's called the Krebs cycle, which generates another 2 ATP. And then, and this is kind of the interesting point, there's another process that you can kind of say happens after the Krebs cycle, but we're in a cell and everything's bumping into everything all of the time. But it's normally kind of viewed to be after glycolysis in the Krebs cycle. And this requires, this requires oxygen. So let me be clear, glycolysis, this first step, no oxygen required, or doesn't need oxygen. This first requires oxygen. So let me be clear, glycolysis, this first step, no oxygen required, or doesn't need oxygen. Oxygen, it can occur with oxygen or without it. Oxygen not needed. Oxygen not needed. Or you could say this is called an anaerobic process. This is the anaerobic part of the respiration. Let me write that down too. Anaerobic. Maybe I'll write it down here. Glycolysis, since it doesn't need oxygen, we can say it's anaerobic. You might be familiar with the idea of aerobic exercise. The whole idea of aerobic exercise is to make you breathe hard because you need a lot of oxygen to do aerobic exercises. So anaerobic means you don't need oxygen. Aerobic means it needs oxygen. Anaerobic means the opposite. You don't need oxygen. So glycolysis, anaerobic, and it produces 2 ATPs net. And then you go to the Krebs cycle. There's a little bit of setup involved here, and we'll do the detail of that in the future. But then you move over to the Krebs cycle, which is aerobic.
It is aerobic. It requires oxygen to be around. And then this produces 2 ATPs. And then this is the part that, frankly, when I first learned, it confused me a lot, but I'll just write it in order the way it's traditionally wrote. Then you have something called, we're using the same colors too much, you have something called the electron transport chain. Electron transport chain. And this part gets credit for producing the bulk of the ATPs, 34 ATPs. And this is also aerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle in the electron transport chain where they need oxygen, instead they go through a side process called fermentation. Fermentation. For some organisms, this process of fermentation takes your byproducts of glycolysis and literally produces alcohol. That's where alcohol comes from. That's called alcohol fermentation. And we, as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. I'll write that down. Lactic acid. That's humans and probably other mammals. Humans. But other things like yeast will do alcohol fermentation. Alcohol fermentation. So this is when you don't have oxygen. It's actually this lactic acid that if I were to sprint really hard and not be able to get enough oxygen, that my muscles start to ache because this lactic acid alcohol comes from. That's called alcohol fermentation. And we, as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. I'll write that down. Lactic acid. That's humans and probably other mammals. Humans. But other things like yeast will do alcohol fermentation. And this part gets credit for producing the bulk of the ATPs, 34 ATPs. And this is also aerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle in the electron transport chain where they need oxygen, instead they go through a side process called fermentation. Fermentation. For some organisms, this process of fermentation takes your byproduct. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle in the electron transport chain where they need oxygen, instead they go through a side process called fermentation. Fermentation. For some organisms, this process of fermentation takes your byproducts of glycolysis and literally produces alcohol. That's where alcohol comes from. That's called alcohol fermentation. And we, as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. I'll write that down. Lactic acid. That's humans and probably other mammals. Humans. But other things like yeast will do alcohol fermentation. Alcohol fermentation. So this is when you don't have oxygen. It's actually this lactic acid that if I were to sprint really hard and not be able to get enough oxygen, that my muscles start to ache because this lactic acid starts to build up. But that's just a side thing. If we have oxygen, we can move to the Krebs cycle, get our 2 ATPs, and then go onto the electron transport chain and produce 34 ATPs, which is really the bulk of what happens in respiration. Now, I kind of said this as an aside that, to some degree, this isn't fair because while these guys are operating, they're also producing these other molecules, or they're not producing them entirely, but what they're doing is they're taking, I know it gets complicated here, but I think over the course of the next few videos, we'll get an intuition for it. In these two parts of the reaction, glycolysis and the Krebs cycle, we're constantly taking NAD, all right, it's NAD+, and we're adding hydrogens to it to form NADH. And this actually happens for 1 molecule of glucose, this happens to 10 NADs, or 10 NAD pluses to become NADHs. And those are actually what drive the electron transport chain. And I'll talk a lot more about it and kind of how that happens and why is energy being derived and how is this an oxidative reaction and all of that, and what's getting oxidized and what's being reduced. But I just wanted to give due credit. These guys aren't just producing 2 ATPs in each of these stages. They're also producing, actually combined, 10 NADHs, which are then used, which each produce 3 ATPs in an ideal situation, the electron transport chain. And they're also doing it with this other molecule, FAD, which is very similar.
but they're producing FADH. Now, I know that all of this is very complicated. I'll make videos on this in the future, but the important thing to remember is cellular respiration. All it is taking glucose and kind of repackaging the energy in glucose and repackaging it in the form of, your textbooks will tell you, 38 ATPs, and sitting in the exam, that's a good number to write. It tends to, in reality, be a smaller number. It's also going to produce heat. Actually, most of it is going to be heat. But 38 ATPs, and it does it through 3 stages. The first stage is glycolysis, where you're just literally splitting the glucose into 2. You're generating some ATPs, but the more important thing is you're generating some NADHs that are going to be used later in the electron transport chain. Then those byproducts are split even more in the Krebs cycle, directly producing 2 ATPs, but that produces a lot more NADHs. And all of those NADHs are used in the electron transport chain to produce the bulk of your energy currency, or your 34 ATPs. We've already learned that cellular respiration can be broken down into roughly 3 phases. Cellular respiration broke down into 3 phases. The first is glycolysis, which literally means the breaking down of glucose. Breaking down of glucose. And then this can occur with or without oxygen. If we don't have oxygen, then we go over to fermentation. We'll talk about that in the future. Go over to fermentation, and in humans it produces lactic acid, and in other types of organisms it might produce alcohol or ethanol. But if we have oxygen, and for the most part we're going to assume that we can proceed forward with oxygen. If there is oxygen, then we can proceed forward to the Krebs cycle. Krebs cycle, sometimes called the citric acid cycle, because it deals with citric acid, the same thing that's in orange juice or lemons. And then from there we proceed citric acid, the same thing that's in orange juice or lemons. And then from there we proceed to the electron transport chain. The electron transport chain, and we learned in the first overview video of cellular respiration that this is where the bulk of the ATP is actually produced, although it uses raw materials that came out of these phases up here. Now what I want to do in this video is just focus on glycolysis. Just focus on glycolysis. And this is kind of a, it's sometimes a challenging task, because you can really get stuck in the weeds, and I'll show you the weeds in a little bit and the actual mechanism, and it can be very daunting. But what I want to do is simplify it for you so you can kind of have the big takeaways, and then we can appreciate, and then maybe when we look at the weeds of glycolysis, we can make a little bit more sense of it. So glycolysis, or really cellular respiration, it starts off with glucose. And glucose, we know its formula. It's C6H12O6. And I could draw its whole structure. It would take a little time, but I'm just going to focus on the carbon backbone. So we can, it is a ring, or it can be a ring, but I'm just going to draw it as 6 carbons in a row. Now, there's 2 kind of important phases of glycolysis that are good to know. One I call the investment phase, and the investment stage phase actually uses 2 ATPs. So it actually uses 2 ATPs. So, the whole purpose of cellular respiration was to generate ATPs, but right from the get-go, I actually have to use 2 ATPs. But I use 2 ATPs, and I'm going to essentially break up the glucose into 2 3-carbon compounds right here that actually also have a phosphate group on them. The phosphate groups are coming from those ATPs. They also have a phosphate group on them. This is often called, well, there's a lot of names for it. Sometimes called P-gal. You really don't have to know this. Or glyceraldehyde, really challenging my spelling skills right here. That's not that important to know. All you have to know is in this first phase, you use 2 ATPs. That's why I call it the investment phase. We use a business analogy, investment phase. And then each of these 2 P-gal molecules can then go into the payoff phase. So in the payoff phase, each of these P-gals turn into pyruvate, which is another 3-carbon, and it's reconfigured. But the process of it going to pyruvate, let me write pyruvate in blue, because this is something that's, at least it's good to know the word, and I'll show you its structure in a second. Pyruvate, sometimes it's called pyruvic acid, same thing, pyruvic acid, and that's essentially the end product of glycolysis. So you start off with glucose in the investment phase, you end up in this phosphoglyceraldehyde, which essentially you broke up your glucose and you put a phosphate on either end of it, and then those each independently go through the payoff phase. So you end up with 2 molecules of pyruvate for every molecule of glucose you started off with. Now you're saying, hey, so that was a payoff phase. What was our payoff? Well, our payoff we got from this. Let me write this down as a payoff phase. This is our payoff phase. And I apologize for the white background. I did it because the mechanism I'm showing you, I copy and pasted it from Wikipedia, and they had a white background, so I just ran with the white background for this video. But I personally at least like the black background a lot better. But this is the payoff phase right here. And so when we go from the phosphoglyceraldehyde to the pyruvate or the pyruvic acid, we produce two things, or I guess we could say we produce three things. We produce each of these p-gals to pyruvates produce two ATPs. So I'm going to produce two ATPs there. I'm going to produce two ATPs there. And then they each produce an NADH. NADH. May I do it in a darker color? NADH. NADH. And of course, they're not producing, they're not producing the whole molecule in a vacuum. Essentially, what they're doing is they're starting with the raw material of an NAD plus. So they start off with an NAD plus, and they essentially reduce it by adding a hydrogen. Remember, we learned a couple of videos ago that you can do reduction as a gain in hydrogen. So the NAD gets reduced to NADH, and then later on, these NADHs are used in the electron transport chain to actually produce ATPs. So the big takeaway here, if I were to kind of write the reaction that we get for glycolysis, is that you start off with the glucose. And you need some NAD plus. And actually, for every mole of glucose, you're going to need two NAD pluses. You're going to need two ATPs. So I'm just writing all the ingredients that we need to start off with. And then you're going to need, these guys are going to be ADPs before we turn them to ADPs. All right, plus 4 ADPs. And then after performing glycolysis, let me write it here. Let me write also that was an AD. ADPs. Let me just rewrite that part right there. 4 ADPs, and then you maybe need 2 phosphate groups because we're going to need 4 phosphate groups. Plus 4. I'll just call them. Sometimes they're written like that, but maybe I'll write it like this: 4 phosphate groups. I'll do 4 phosphate groups like that, and then once you perform glycolysis, you have 2 pyruvates. 2 pyruvates or glucose. You're going to need 2 NAD pluses. You're going to need 2 ATPs. So I'm just writing all the ingredients that we need to start off with. And then you're going to need, these guys are going to be ADPs before we turn them to ADPs. All right, plus 4 ADPs. And then after performing glycolysis, let me write it here. Let me write also that was an AD. ADPs. Let me just rewrite that part right there. 4 ADPs, and then you maybe need 2 phosphate groups, or we're going to need 4 phosphate groups.
You still write it the same way.
4, I'll just call them, sometimes I write it like that, but maybe I'll write it like this: 4 phosphate groups. I'll do 4 phosphate groups like that, and then once you perform glycolysis, you have 2 pyruvates. You have 2 NADHs. The NAD has been reduced. It gained a hydrogen, RIG, oil rig. Reduction is gain in electron, but in the biological sense, we think of it gaining the hydrogen because hydrogen is very non-electronegative, so you're hogging its electrons. You gained its electrons. So 2 NADHs, and then plus these 2 ATPs get used in the investment phase. That's why I kind of wrote them a little separately. So these 2 get used. So then you're left with 2 ADPs, and then these guys essentially get turned into ATPs. So plus 4 ATPs. I guess we didn't need 4. We only needed a net of 2 phosphate groups because 2 jump off of here, and then we need a total of 2 more to get 4 jumping on there. But the big picture is you start with glucose, you end up with 2 pyruvates, you use up 2 ATPs, you get 4 ATPs, so you have a net of 2 ATPs formed. Let me write that very big. Net, what you get out of glycolysis is 2 ATPs, you get 2 NADHs that can each later be used in the electron transport chain to produce 3 ATPs, and you get 2 NADHs, and you get 2 pyruvates, which are going to be re-engineered into acetyl-CoAs that are going to be the raw materials for the Krebs cycle. But these are the outputs. These right here are the outputs of glycolysis. So now that we have that big picture, let's actually look at the mechanism, because this is a little bit more daunting than you see it here, but we'll see the same themes that I just talked about. We're starting with the glucose right there. It is a 6-chain, it's in a circle, it's in a ring, 1, 2, 3, 4, 5, 6 carbons. I could write it like that, just to make it a huge oversimplification. Goes through a few steps. I use an ATP here, so that's, let me do that in a color. We do it in orange whenever I use an ATP. I use 1 ATP there, I use 1 ATP there, and just like I told you, they have a slightly different name for it, but this is the phosphoglyceraldehyde right here. They call it glyceraldehyde 3-phosphate. It's the exact same molecule. But as you can see, just when I drew it very roughly before, you've got 1, 2, 3 carbons there. 1, 2, 3 carbons there, and it also has a phosphate group on it. The phosphate group's actually attached to the oxygen, but just for simplification, I draw the phosphate group just like that. And I showed that right here. This was the phosphoglyceraldehyde right here. This is the actual structure up here, but I think sometimes we look at the structure, it's easy to miss the big picture. And there are two of these. They kind of say that you can go back and forth with this other kind of isomer of this. But the important thing is that you have two of these compounds that are now 3 carbon compounds. Glucose has been split, and now we're ready to enter the payoff phase. And remember, you have 2 of these compounds. 2 of these compounds right here. That's why when they drew this mechanism, they wrote ×2 right there, because the glucose has been split into 2 of these molecules. So each of the molecules are now going to do this right here. And for each of the glyceraldehyde 3-phosphates, or PGALs, or phosphoglyceraldehydes, we can look at the mechanism. We can look at the mechanism and say, okay, look here, there's going to be an ADP turning into an ATP there. So this is +1 ATP. And then we see it again happening here on our way to pyruvate. On our way to pyruvate right there. Then we have another +1 ATP. So for each of the PGALs, or the phosphoglyceraldehydes that were produced, we're producing 2 ATPs in the payoff phase. Now there were 2 of these, so total for 1 glucose, we're going to produce 4 ATPs in the payoff phase. So in the payoff phase, 4 ATPs. In the investment phase, we used 2 ATPs. So total net ATPs directly generated from glycolysis is 2 ATPs, right? 4 gross produced, but we had to invest 2 in the investment phase. And then the NADs, the NADHs, we see right here. For each phosphoglyceraldehyde, or glyceraldehyde 3-phosphates, or PGALs, whatever you want to call them, at this stage right here, you see that we are reducing NAD+ to NADH. So this happens once for each of these compounds, and obviously there are 2 of these. Glucose got split into 2 of these guys, so 2 NADHs are going to be produced, and later these are going to be used in the electron transport chain to actually each produce 3 ATPs. And then finally, when everything is said and done, we're left with the pyruvates, and it's nice that they made it nice and big. We can take a look at what a pyruvate looks like, and just as promised, we can look at all the oxygen bonds and all of that, but it is a 3-carbon structure, has a 3-carbon backbone. So the end result is that the glucose got split in half. It got oxidized. Some of the hydrogens got stripped off of it. As you can see, there's only 3 hydrogens here. We started off with 12 hydrogens in glucose, and now carbon is bonding more strongly with oxygen, so it's essentially having its electrons stolen by the oxygens, or hogged by the oxygens. So carbon has gotten oxidized in this process. There's going to be more oxidation left to do. And in the process, we were able to generate 2 net ATPs, and 2 NADHs that can later be used to produce ATPs. Anyway, hopefully you found that helpful.
All right, let's take a 5-minute here. Let's take a 5-minute. Let's just take a 10-minute. Let's take a 10-minute. We'll come back. We'll finish this, and then I have to. I'll do it on stream, but I have to read the same thing again, anyways, and then also do a little bit of action potentials for the physio class. So we'll do that, and then and I'll take the quiz for it, and then just go from there. And then finish it up, right? Just finish it up. And then today, maybe tomorrow, do the bio book. And actually, I think we should do bio and then biochem before Gen Chem. All right, cool. Very nice. All right, I'll be back. Yeah. Okay. Let's do it. Oh, guess what I got? I got some milk. Pretty cool, huh? Yeah. So good. Life is good. Life is great. That's okay. That's okay. Life is still good. Life is still great. So good. So good. Yep. We did 315 yesterday. We could try to do 316 today. I don't know what they're doing. Okay. That makes sense. Glycogenesis is stimulating glycogen synthase, increasing glycogen storage. Blood, that. When blood glucose levels are low, glucagon will stimulate glycogenolysis, breakdown of glycogen into glucose. Doing so by activating glycogen phosphorylase. So cleaves alpha 1,4 glycosidic bonds to release glucose 1 phosphate. And then the glycogen is okay. Glycogenolysis. Yeah.
The body is able to maintain a very narrow and constant range of blood glucose in our body. So notably about 60 to 150 milligrams of glucose per deciliter of blood. And it's not important that you know this exact number, but what I think is significant, what I think is pretty fascinating is that our body is able to maintain a very narrow and constant range of blood glucose in our body. So notably about 60 to 150 milligrams of glucose per deciliter of blood. And it's not important that you know this exact number, but what I think is significant is that in contrast to something like free fatty acids, for example, which we'll talk about in fatty acid metabolism, fatty acids can range almost tenfold depending on the needs of the body. So they can either be really, really high or really, really low. But glucose always stays within a very constant range, and say blood glucose level here. And it's important that this is a very constant range, of course, because there are some tissues in our body, such as our brain and some of the cells in our eyes and our kidneys and even our red blood cells, that rely on glucose nearly exclusively to produce ATP. So remember, once glucose is in the blood, it can be used by any of the cells in our body through process of cellular respiration to produce ATP. So remember, the three big steps of cellular respiration are first glycolysis, the breakdown of glucose, and then the glucose goes to the Krebs cycle, where it undergoes some more oxidation to release all of that energy in the glucose molecule. And finally, the byproducts of the glycolysis and Krebs cycle go to the electron transport chain, which is able to produce ATP in bulk amount. So how does our body keep this blood glucose in such a narrow range and constant range in our body? So our body is able to do this differently depending on kind of what state the body is in. So the body can be either in the fed state, or our body can be in something that we call a fasted state. So you can imagine kind of the fed state.
Yep. Oh, my back. You're losing weight. Not really. Nope. Really? I think we'll do like a lower leg workout today. Or just leg workout. Lower leg workout today. Something. We'll do something. We'll figure it out.
This ATP in bulk amount. So how does our body keep this blood glucose in such a narrow range and constant range in our body? So our body is able to do this differently depending on kind of what state the body is in. So the body can be either in the fed state, or our body can be in something that we call a fasted state. So you can imagine kind of the fed state as being just after you've eaten a meal. So let's say you've eaten a chocolate chip cookie. The glucose that has been broken up in your GI tract can then be used to directly contribute to this blood glucose level. And then, of course, the glucose can be used by our cells. Now, in the fasted state, so imagine that this is basically all the times you're not eating, the body has come up with kind of two different ways to regulate blood glucose levels. So now remember that in a fasting state, our body needs a way to essentially pump glucose into the blood to keep it at this level, essentially to replace the glucose that's being used up by our cells because we don't have this constant intake from, a chocolate chip cookie. So in this case, our body has glycogen, which is a polymer or kind of essentially a string of glucose molecules that it essentially stores away. And our body actually kind of ingeniously makes this glycogen by using some of that glucose that is dumped into our body during the fed state. So by using some stores away. And our body actually kind of ingeniously makes this glycogen by using some of that glucose that is dumped into our body during the fed state. So kind of in anticipation of knowing that it's not always going to get glucose from eating, it kind of preserves some of it in this glycogen molecule. And most of this glycogen molecule is actually, just as a fun fact, located in your liver, which is why your liver is very, very important for carbohydrate metabolism. And so in times of fasting, our body can actually go ahead and break down this glycogen into the individual glucose molecules, which then can be used to keep our blood glucose levels constant. So unfortunately, it turns out that this mechanism of breaking down glycogen only lasts for about 10 to 18 hours in our body. That is to say that after 10 to 18 hours, we've essentially used up all of our glycogen stores and we need to eat another meal to kind of build those glycogen stores back up. So you can imagine during an overnight fast, for example, so, when you go to sleep and, it's usually about, hopefully 8 to 10 hours. And so you can imagine there's a point during the day when your body needs another way of producing glucose. And so our body has come up with a second way called gluconeogenesis, which is indeed the topic of this video. And gluconeogenesis is exactly what its name implies. It is the genesis or creation of new glucose. Now, it's actually quite fascinating just to kind of think about this for a moment. What we're saying in gluconeogenesis is essentially our body is taking precursor molecules that are from a non-carbohydrate source. So essentially it looks at what it has lying around, and notably, most commonly, it uses amino acids in our body, as well as a molecule called lactate, which is produced as kind of a byproduct, oftentimes in exercising muscle cells. And it takes kind of these precursor molecules and reconfigures them to produce glucose. And it's this glucose that can then be used to be dumped into our blood to maintain this constant blood glucose concentration and a constant supply of ATP for our tissues. So now that you kind of have a big picture of carbohydrate metabolism and where gluconeogenesis fits in, let's go ahead and talk about, as promised in this video, about this metabolic pathway, gluconeogenesis. So in order to do this, I think most effectively, it's actually important to revisit glycolysis briefly. So I'm going to go ahead and bring up the reaction diagram that was used to explain glycolysis in a previous video. So just to orient you, remember that glycolysis begins with glucose up here, and glucose is broken down in a series of steps. Most notably at one point it's broken down into this 3-carbon molecule, glyceraldehyde 3-phosphate, and then it is broken down even further and reconfigured, releasing some ATP and NADH along the way, and ultimately forming this molecule pyruvate. And pyruvate is a very important molecule because it can continue to the Krebs cycle, where it can be further oxidized to produce more NADH that can be used by the electron transport chain to produce ATP. All right, so that was kind of a big mouthful, but just remember, big picture, glycolysis breaking down glucose into pyruvate. Turns out, the way I like to think about gluconeogenesis is that the goal of gluconeogenesis is to produce glucose. And so essentially the way I like to think about it is that gluconeogenesis is almost the exact reverse pathway of glycolysis. That is to say, we start at this end of the reaction pathway, we start with pyruvate, and we essentially go funnel back.
Wow.
The opposite direction through all of these reactions to produce glucose. Now, the key word is that it's almost the exact reverse of glycolysis, and it's almost the reverse because I want to call attention to these orange arrows. So note that there are kind of 3 orange arrows. So one from glucose to this molecule glucose 6-phosphate, another one here, and then one at the very end, which converts the last molecule to pyruvate. And what's important to note about these reactions in glycolysis is that unlike the other bidirectional black arrows that are used in most of the reactions, these orange arrows are unidirectional. What they're trying to indicate is that these 3 reactions are irreversible. In other words, they have a negative, if we plot a fancy term from chemistry, they have a negative delta G value, or a negative Gibbs free energy, which means that if we were to reverse these particular reactions, we would have to flip the sign, right? And so these negative delta G values would become positive. And that's problematic, right? Because we know that in order for any biological reaction to occur, we must have a negative delta G value. So, for reaction to occur, and that's problematic, right? Because we know that in order for any biological reaction to occur, we must have a negative delta G value. So, our body has come up with a bit of a compromise. Our body has essentially said, okay, look, we have 1, 2, 3, 4, 5, 6, 7 reactions with these kind of bidirectional black arrows that are essentially reversible. That is to say, they have a delta G value that's near 0. And so they can go either direction. So our body says, okay, we'll keep those 7 reactions. But in going from pyruvate back to glucose, we have to come up with a different reaction pathway for the 3 steps that are irreversible. So that's exactly what our body did. And in fact, that's what I'll go ahead and review in the remainder of this video. But essentially, you can say, with those 3 steps in mind, we're just performing the reverse of glycolysis.
Feels like it kind of ended a little bit short. No?
All right, so in the previous video, we talked about the big picture of gluconeogenesis, or the creation of a new glucose. And I brought up this diagram of glycolysis, and I said to you, essentially, glycolysis, which starts off with glucose that you can see in the kind of top middle left and is broken down, oxidized into pyruvate, the molecule kind of in the top left there. I said that this pathway is essentially the reverse of gluconeogenesis. That is to say, we want to start off with pyruvate, and we want to essentially reverse the pathways to produce glucose, which we can pump into our blood in times of fasting. But notably, in this video, I want to get a little bit more detail-oriented and talk about kind of 3 unique reactions to gluconeogenesis that overcome the 3 irreversible steps indicated by the orange arrows in this diagram of glycolysis. All right, so let's talk about the first roadblock that we need to overcome, which is in the conversion of pyruvate to phosphoenolpyruvate. So remember, it can't just use pyruvate kinase to reverse this reaction. So it actually uses an entirely separate set of enzymes and pathway to get to phosphoenolpyruvate. And the first step is the conversion of pyruvate to a molecule called oxaloacetate, which is abbreviated usually as OAA. And this is a molecule that you will meet when you learn about the Krebs cycle. It's a intermediate in one of those steps. And notably, this is why amino acids are able to be used to produce glucose, because amino acids, once they're broken down, can be actually converted to oxaloacetate. And from there, of course, they can continue down this pathway. Notably, another precursor molecule that I mentioned before, lactate, can be interchangeably produced from pyruvate or going from lactate to pyruvate. So that's another way that that metabolite contributes to gluconeogenesis. But back to oxaloacetate. So once this is produced, oxaloacetate can then be catalyzed to form phosphoenolpyruvate. They can continue down this pathway. Notably, another precursor molecule that I mentioned before, lactate, can be interchangeably produced from pyruvate or going from lactate to pyruvate. So that's another way that that metabolite contributes to gluconeogenesis. But back to oxaloacetate. So once this is produced, oxaloacetate can then be catalyzed to form phosphoenolpyruvate using another enzyme. And the names of these enzymes aren't terribly important, but I will mention them because they do sometimes come up. So the first step involves an enzyme called pyruvate carboxylase. And notably, kind of one way that I remember this is because oxaloacetate is actually a 4-carbon molecule. So pyruvate, recall, is a 3-carbon molecule. So this carboxylase enzyme is essentially adding another carbon through a carboxy group to this oxaloacetate molecule. And then this oxaloacetate molecule is converted back to a 3-carbon intermediate by an enzyme called PEP, which stands for phosphoenolpyruvate for short, PEP. Converted by PEP carboxy kinase. And of course, kinase involves the phosphorylation of something. And so we know actually intuitively that because gluconeogenesis is an anabolic process, we're building something up, it requires energy. And energy comes usually in the form of ATP. And it turns out that this first step of the reaction does involve ATP. So I'm going to say plus ATP. And the second step, where this kinase is involved, also involves energy in the form of GTP, which is pretty much the equivalent of ATP. But of course, we have this G, which is just guanine, a different nucleotide base. But this, of course, we know the energy is derived from these phosphate groups. So essentially, we can think of these things as being the same. All right, so 1 roadblock down and 2 left to go. So once we have our phosphoenolpyruvate, we're good to go. We shuttle down this pathway until we hit our next roadblock, which is in the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. So the way our body does this is to actually use a different enzyme. So normally, phosphofructokinase is used to convert fructose 6-phosphate to fructose 1,6-bisphosphate. And instead, in gluconeogenesis, going the opposite direction, the body uses an enzyme called fructose 1,6. So we're talking about the same molecule here. But instead of kinase, we're using a biphosphatase. It's kind of a mouthful, but just recognize that a phosphatase is the exact opposite of a kinase. So whereas kinases involve phosphorylation, usually using phosphate groups from molecules like ATP, a phosphatase takes these phosphates away. So naturally, it makes sense that if a kinase is used in going from fructose 6-phosphate to fructose 1,6-bisphosphate, we'd want to take a phosphate group off, going the opposite direction. So that's exactly what we do. Now, one kind of point of confusion that kind of I had when I was learning this was, we always learn that the enzyme of a reaction can't really change whether a reaction is irreversible or not. So I want to recall in going from fructose 6-phosphate to fructose 1,6-bisphosphate, we'd want to take a phosphate group off, going the opposite direction. So that's exactly what we do. Now, one kind of point of confusion that kind of I had when I was learning this was, we always learn that the enzyme of a reaction can't really change whether a reaction is irreversible or not. So I want to point out that it's not just an enzyme that we're switching out and going and kind of circumventing this irreversible reaction, because that really wouldn't do anything. If a reaction has a negative delta G value, it will always have a negative delta G value. And changing the enzyme won't change the delta G value. Remember, kinetics and thermodynamics are separate entities. And so I want to point out to you that it's really this entire reaction pathway that's changing. And notably, remember that ATP in this step is hydrolyzed to ADP. And this reaction is coupled normally to the phosphorylation with the enzyme phosphofructokinase. But this hydrolysis of ATP is absent in gluconeogenesis. So we can essentially think about this switch in enzyme as really encompassing a larger change in the entire pathway going from fructose 1,6-bisphosphate to fructose 6-phosphate. So I hope that's clear. So now that we have gotten past this second roadblock, we continue down until we hit our final and last roadblock and going from glucose 6-phosphate to glucose. And again, essentially just like we did for our previous reaction, our body has come up with a different reaction pathway involving a different enzyme. So normally you remember hexokinase is used in converting glucose to glucose 6-phosphate, but our body uses a different enzyme. In this case, I'm going to write it out here. It uses the enzyme glucose 6. And remember, if we used a kinase, we have to be using we're using a phosphatase. So glucose 6 phosphatase, which will remove this phosphate group from the glucose to remove this phosphate group from the glucose 6 phosphate to form the glucose. Now, just as a fun kind of intriguing fact, it turns out that some people are actually missing this enzyme glucose 6 phosphatase. So can you imagine what would happen if they're missing this enzyme? And indeed, without this enzyme, we can't produce glucose. So it's kind of sad, right? Because those individuals can perform all of these reactions leading up to glucose 6-phosphate, but it can't ever produce glucose. And what's actually even more interesting about this enzyme is this enzyme is also used in the breakdown of glycogen, which, if you remember, we mentioned earlier as that polymer of glucose that's used kind of as a first line dumping mechanism for glucose into the blood during our fasting state. So not only are these individuals unable to produce glucose using gluconeogenesis, but they're also unable to break down their glycogen, which means that they're severely hypoglycemic. So without this enzyme, I'm going to have an arrow and cross it out. Without this enzyme, individuals are hypo or low in glucose. And of course, this is a very life-threatening condition because our body needs glucose to survive. All right, so just kind of as one final word, I want to say that when I was first learning about gluconeogenesis and glycolysis, I wanted to memorize all of the names of all the molecules and all the enzymes. And speaking of enzymes, I think I spelled one wrong here. This is supposed to be 1,6-bisphosphate. But despite that, really, I guess the key point I want to say is that, these names can be important when we're talking about specific diseases like this deficiency of glucose 6-phosphatase. But conceptually, it's, I think, enough to realize at this point that gluconeogenesis and glycolysis are essentially opposites, minus these 3 irreversible steps for which our body has created 3 unique reaction pathways for which gluconeogenesis can occur. So that's kind of the big takeaway from this video. At its most simplistic level, regulation of metabolic pathways inside of the body is really just a fancy word for a balancing act that's occurring in the body. So to illustrate this, I have a seesaw, and we've been learning about 2 metabolic pathways, glycolysis, which is the process of breaking down glucose into pyruvate, and gluconeogenesis, which is essentially the opposite, in which we start out with pyruvate, and through a little bit of a different route, we end up back at glucose. And when we're talking about the regulation of these particular pathways, we're essentially asking ourselves, when is glycolysis the predominant pathway, and when is gluconeogenesis the predominant pathway? The body wants to make sure that we either have a net breakdown of glucose in the case of glycolysis, or that we have a net production of glucose in the case of gluconeogenesis. So now the next question is, how does the body accomplish this balancing act? And to answer this question, the way I like to think about it is to think about it along a spectrum. There are very fast-acting forms of regulation that take place on the order of seconds, and there are very slow forms of regulation that can take up to hours or even days to occur. So let's talk about each of these in a little bit more detail. The major principle that helps me understand fast-acting forms of regulation is a good old principle from general chemistry, Le Chatelier's principle. So if you remember, Le Chatelier's principle talks about anything that's in equilibrium, and it says that if there's any change to this equilibrium, let's say more products are added or reactants are taken away, the equilibrium will adjust to essentially counter that change and return the system back to equilibrium. So what does this mean in the context of metabolic pathways like glycolysis and gluconeogenesis? So let's remind ourselves that in glycolysis, glucose is converted to pyruvate through several reactions that are kind of all in equilibrium with one another. And so we can essentially think about this metabolic pathway as a series of equilibria. And so imagine, for example, if we had an influx of glucose. Let's say we've just eaten a big meal and a huge bunch of glucose has entered our body and our bloodstream. What will happen to this equilibrium? Well, we can return to Le Chatelier's principle and say that if we have a rise in glucose, it will essentially push this entire equilibria towards the production of pyruvate. And so you can see that in this example, Le Chatelier's principle allows equilibrium to adjust within seconds to just a simple influx of glucose to promote glycolysis. Le Chatelier's principle also applies to gluconeogenesis. So remember that in gluconeogenesis, something unique starts to happen after blood glucose levels have been low for a while. Amino acids begin to break down and form this metabolite called oxaloacetate. And remember that oxaloacetate plays this unique role in the conversion of pyruvate back to glucose, which occurs in gluconeogenesis. Remember that it's kind of this intermediary. So pyruvate is converted to oxaloacetate and then essentially re-enters the equilibria to form glucose. So you can imagine that if we have an influx of oxaloacetate, the equilibria will be pushed towards the opposite direction, that is, towards the production of glucose. Now, in addition, I want to briefly mention another form of fast-acting regulation, which is called allosteric regulation. So what is allosteric regulation? Recall that all metabolic pathways have unique enzymes that catalyze or facilitate each step of the reactions along the metabolic pathways. So you can imagine that if we have an enzyme here, I'm just drawing a simple structure, it has what's called an active site, where it actually binds the substrate of interest. So it binds the, let's say, glucose molecule here. But in addition, there are also molecules within the cell that we call allosteric regulators. And these, by definition, bind to a portion of the enzyme that is not the active site. So let's say we have an allosteric molecule that binds to a separate portion, like right here. Now, this allosteric molecule can have one of two effects. We say that allosteric molecules can be inhibitory, that is, by inhibiting enzymes, inhibit the pathway that utilizes those enzymes, or these allosteric interactions can be positive, that is, promote the action of enzymes and therefore promote the overall reaction in which those enzymes are involved. So to put this in context with glycolysis and gluconeogenesis above, it turns out that ATP is actually a big allosteric regulator of one of these two pathways. So recall that gluconeogenesis requires ATP, or a net amount of ATP, to produce glucose. It's an anabolic building up pathway. On the other hand, in glycolysis, there is a net release of ATP and the oxidative breakdown of glucose. And so if we have a lot of ATP in a cell, think about for a moment which of these two pathways would be favored. Indeed, gluconeogenesis would probably be favored because it requires ATP.
Hold on.
So there is a net release of ATP and the oxidative breakdown of glucose. And so if we have a lot of ATP in a cell, think about for a moment which of these two pathways.
If you had a lot of ATP, we're gonna go use them up. Use them up, and we're gonna go back to glucose. We're gonna go back to glucose. Yeah, that makes sense. That makes sense.
Would be favored. Indeed, gluconeogenesis would probably be favored because it requires ATP.
Yes.
On the other hand, if there's a lot of ATP, that could.
You said if there's a lot of ATP, we're gonna go to gluconeogenesis. Yeah.
Kind of a sign to the cell to say,
Hey, stop breaking down glucose.
hey, we don't need to perform as much glycolysis because we already have enough ATP available. And it does turn out that ATP is actually an allosteric regulator of a couple enzymes in glycolysis.
I did not know that. I did not know that. Thank you, ma'am.
Say, hey, we don't need to perform as much glycolysis because we already have enough ATP available. And it does turn out that ATP is actually an allosteric regulator of a couple enzymes. In glycolysis, and specifically, it's a negative allosteric regulator or an inhibitor of these couple enzymes. Essentially, it's putting the brakes on glycolysis and saying we have enough energy and we don't need to produce anymore. On the other hand, it turns out that there is also a molecule, AMP, in the body, which is basically a sign that the cell has used up to produce anymore. On the other hand, it turns out that there is also a molecule, AMP, in the body, which is basically a sign that the cell has used up all of its ATP. In other words, ATP has been dephosphorylated and turned into AMP, which is a sign that the cell is running out of ATP. So if the cell is running out of ATP, the cell probably won't want to be performing energy-requiring processes such as gluconeogenesis. And indeed, AMP is a negative allosteric regulator of one of the enzymes in gluconeogenesis. All right, so that kind of finishes up our discussion of fast-acting forms of regulation. So now let's talk briefly about slow-acting forms of regulation. So these types of regulation often take advantage of transcriptional changes within the cell. So what do I mean by that? So let's first remind ourselves what transcription is. So remember that transcription is the process of taking DNA and making an mRNA transcript and then translating this in the cytosol of the cell to a protein product. And when we're talking about proteins, oftentimes we're talking about enzymes. I'm just going to go write that here since it's relevant for our discussion. And so you can imagine, for example, that this might be very useful if the organism is in a long-term fasting state. It will want to essentially upregulate the transcription of enzymes that promote something like gluconeogenesis so that it can dump glucose into the blood. And notice here that, even visually as it's implied here, this process of going from DNA to mRNA to enzymes is going to take much longer than, a simple Le Chatelier or allosteric regulation. And so that's why this process is more of an adaptive process that allows the organism to adapt to more kind of long-term changes that it experiences in its environment. Now, finally, I want to add in one more form of regulation between fast and slow-acting regulation, which is called hormonal regulation. So what is hormonal regulation? Well, it's exactly what it sounds like. It's the ability for the body to essentially produce specific hormones, which are simply molecules that travel in the blood to regulate whether glycolysis or gluconeogenesis is on or off. And the two hormones that the body uses to regulate glycolysis and gluconeogenesis, and pretty much actually all metabolic pathways, are insulin and another hormone called glucagon. And depending on whether there is more insulin or more glucagon, the body will be more likely to do glycolysis or more likely to do gluconeogenesis. So let's talk about how that decision is made. Now, hormones like insulin and glucagon are usually released by the body whenever the body deviates from a particular set point. Now, in the case of regulation and metabolism, the set point that we're interested in is the blood glucose level. And if we return back to our analogy here, this seesaw here, this kind of pivot point we can kind of think about as our set point, the blood glucose level. It's a specific amount of glucose that the body wants to have in the blood at all times. Now, to get more specific, if the blood glucose level rises, it actually stimulates the body to release the hormone insulin. And if the blood glucose levels decrease, it stimulates the body to release the hormone glucagon. And so with that in mind, take a moment to think about which hormone, insulin or glucagon, promotes glycolysis and which of these two hormones promotes gluconeogenesis. Basically, this is
well, wait. Yeah, I'd probably say glucagon for gluconeogenesis and glycolysis for insulin.
It's actually a macro application of Le Chatelier's principle, right? If we have too much blood glucose level, we want to get rid of it. How do we get rid of it? We break it down. And so indeed, insulin promotes glycolysis. On the other hand, when blood glucose levels are low, we want to return the equilibrium to normal. We want to pump more glucose back into the blood, and we know that gluconeogenesis can accomplish that for us. And so glucagon indeed promotes gluconeogenesis. Now, briefly at the end, I want to talk about why I decided to put hormonal changes between fast and slow-acting forms of regulation. So to talk about this, we need to understand a little bit how hormones interact with target cells. So cells in our body have particular receptors that will bind to the hormones that are floating around in the bloodstream. So once these receptors bind to a particular hormone, whether it be insulin or glucagon, it actually causes a series of particular reactions to occur inside of the cell to modify, oftentimes, enzymes that are involved in metabolic pathways. So I'll just abbreviate that with the letter E. And these modifying reactions that occur in the cell are oftentimes phosphorylation reactions. That is either the gain or the loss of a phosphate group, oftentimes on an enzyme in a metabolic pathway. And so with that in mind, you can appreciate how modification by phosphorylation is a lot faster than starting with a DNA transcript and then going to mRNA and then translating into enzymes, but it is indeed a bit slower than kind of a second-to-second Le Chatelier and allosteric regulation that can occur in the cell as well.
Right.
When talking about carbohydrate metabolism,
Pentose phosphate pathway. All right. Yeah, not back yet, but we are kind of almost done. Um, yeah, probably we're gonna have to end it. This will be. We can leave this for tomorrow. Plus, reading the book. But actually, we're gonna hit biochem next. Let me clean up the desk real quick. Hold on, guys. Give me 5 minutes. Like, maybe 5 minutes. Let me take out the trash, and then we can finish this. I have to finish the assignment for physio as well. Okay.
Mention the pentose phosphate pathway. So where does the pentose phosphate pathway fit into the breakdown of glucose? So let's go ahead and review the breakdown of glucose as we normally kind of usually conceive of it as. So we go ahead and start out with glucose, which I'm drawing here just symbolizing with a 6-carbon sugar backbone. And we usually imagine that glucose begins to be broken down in the cytosol of the cell through a series of reactions that we call glycolysis. And then, of course, it goes through the Krebs cycle in the mitochondria, also known as the TCA cycle. And then finally, it goes to the electron transport chain in the mitochondria to produce ATP. So that's kind of usually the end product we think of when we think about breaking down glucose. But the pentose phosphate pathway is kind of a unique pathway because it turns out that in this pathway, no ATP cycle like glucose has. So we go ahead and start out with glucose, which I'm drawing here just symbolizing with a 6-carbon sugar backbone. And we usually imagine that glucose begins to be broken down in the cytosol of the cell through a series of reactions that we call glycolysis. And then, of course, it goes through the Krebs cycle in the mitochondria, also known as the TCA cycle. And then finally, it goes to the electron transport chain in the mitochondria to produce ATP. So that's kind of usually the end product we think of when we think about breaking down glucose. But the pentose phosphate pathway is kind of a unique pathway because it turns out that in this pathway, no ATP is consumed or produced. That's kind of unique to point out. So where does it fit into this overall pathway? It turns out that the linear way I've written cellular respiration is actually only partly true. It's a great way to conceptualize it, but there are many branches or kind of side reactions that are taking place almost simultaneously with the breakdown of glucose. And the pentose phosphate pathway is one of these. So it turns out that as glucose begins to go through glycolysis, some of it is shunted away to become the pentose phosphate pathway. So glucose continues to be broken down, but it continues to be broken down to produce different products than it would if it continued through glycolysis and Krebs and into the electron transport chain. So as you can see, I've written pentose phosphate pathway kind of suggestively by highlighting pentose and phosphate in different colors to point out to you that there are 2 primary products in this pathway. So the first is the production of a 5-carbon pentose sugar. So pentose is just another word for 5-carbon sugar. And the particular name of this sugar is ribose 5-phosphate. And this sugar, so the 5-carbon sugar, I'll go ahead and draw that to remind us of that, is an important substrate in producing DNA and RNA. So if you remember, DNA and RNA contain nucleotides, and the nucleotides contain a nitrogenous base, a phosphate group, and a 5-carbon sugar. So in the case of DNA, it's deoxyribose, and in RNA, it's just ribose. But in either case, this ribose 5-phosphate is an important precursor to creating DNA and RNA, so quite a crucial molecule. Now, the second primary product of this reaction, as this phosphate nicely implies, is a phosphorylated molecule that is usually abbreviated as NADP, P standing, of course, for the phosphate in this molecule, H. NADPH. So this is not to be confused with the NADH, which, if you recall, go ahead and actually draw that in here. If you recall, NADH is actually produced in cellular respiration during the breakdown of glucose. So this produces NADH, which of course contributes electrons to the electron transfer chain. So of course, the question you might have in your mind is, how is NADH different from the easily confused NADPH? Because they sound like similar molecules, and in many ways they are. So they actually both exist in pairs inside the cell. So NAD plus, we know, is interconverted with NADH, and NADP plus is interconverted with NADPH. So of course, the H forms of these molecules are the reduced form of these molecules, and the plus or NADH, and NADP plus is interconverted with NADPH. So of course, the H forms of these molecules are the reduced form of these molecules, and the plus or oxidized form of these molecules are the NAD plus and NADP plus. But what's different about these two pairs of molecules is the relative amount of the reduced form and the oxidized form inside the cell. So just to give you a sense of that, the ratio of NAD plus to NADH is about 1,000. In other words, if you took the amount of NAD plus and divided it by the amount of NADH in the body, you would have about 1,000 times more NAD plus. On the other hand, if you took the amount of NAD plus, NADP plus, divided by the amount of NADPH, you would get 0.1. So essentially what this is telling us is that there's a lot of NAD plus in the body and a lot of NADPH in the body, but not much of NADH or NADP plus. And knowing this actually helps me remember and differentiate between the role of NADH and NADPH inside the body. So first, I reason out to myself that if there's a lot of NAD plus present in the body, most of the NAD plus will want to accept electrons. And of course, the biggest role in accepting electrons comes in the breakdown of glucose and producing NADH. So that makes sense. On the other hand, the primary role of NADPH, which is what we have the majority of, is to donate electrons. I'm going to go ahead and write that here. So the biggest role of NADPH in the body is to donate electrons. And that, of course, would not be very helpful in breaking down glucose, right? Because the breakdown of glucose donates electrons. It doesn't accept them. Now, I will remind you that donating electrons is really important in anabolic reactions. So remember that anabolic reactions involve building up molecules, such as in the synthesis of fatty acids, for example. And so NADPH plays a vital role in kind of providing this reducing power, so to say, for these anabolic reactions. In addition, I'll briefly mention that NADPH also uses its reducing power, its ability to donate electrons, to maintain the store of antioxidants inside the body. So, kind of an ironic part about having oxygen as a requirement for cellular respiration is that some of this oxygen can become really reactive if it gains an extra electron. And so the role of kind of some of the molecules in your body are to serve as antioxidants to kind of trap these reactive oxygen species from reacting with important things in your body, like DNA or proteins. And so once they do that, of course, some of these antioxidant molecules, in the process of reacting with a reactive electron-rich oxygen molecule, become oxidized. And so, of course, NADPH can come in and save the day by donating electrons to reduce the oxidized form of these antioxidants back into the reduced form so that they can again react with any rogue reactive oxygen species. All right, so now we're ready to look at the pentose phosphate pathway in more detail. So I'm going to go ahead and bring up a diagram of how the pentose phosphate pathway is usually represented in most textbooks. And this is a lot of detail, admittedly, and I don't want you to get lost in the details. So I'm going to try and break it down and hone your attention to the most important details to take away from this. So the first of these important details is to note that there are two big phases of the pentose phosphate pathway. So the first is called the oxidative phase, and the second is called the non-oxidative phase. And, as the name implies, oxidative phase, we're oxidizing. So remember that breakdown of glucose, breakdown of carbohydrates is an oxidative process in general. And in this phase, the big idea here is that we are producing NADPH. So that is the big product of the oxidative phase. So we actually start out with glucose 6-phosphate here. So just note that we start off with this molecule here, which I'll remind you is one of the first metabolites that's produced in glycolysis. So this is essentially shunted from glycolysis, which of course starts out with glucose. The glucose enters glycolysis, and some of it will continue through cellular respiration, but the other part of the glucose will then be shunted through this glucose 6-phosphate into the oxidative phase of the pentose phosphate pathway. And glucose 6-phosphate is then broken down in a series of steps, which aren't entirely important, but the key idea here is that you're producing NADPH along the way. Now, the non-oxidative phase starts with this molecule called ribulose 5-phosphate. And it's really not important to know, except for the fact that it kind of sounds like ribose 5-phosphate, right? Which I mentioned that we are producing NADPH. So that is the big product of the oxidative phase. So we actually start out with glucose 6-phosphate here. So just note that we start off with this molecule here, which I'll remind you is one of the first metabolites that's produced in glycolysis. So this is essentially shunted from glycolysis, which of course starts out with glucose. So glucose enters glycolysis, and some of it will continue through cellular respiration, but the other part of the glucose will then be shunted through this glucose 6-phosphate into the oxidative phase of the pentose phosphate pathway. And glucose 6-phosphate is then broken down in a series of steps, which aren't entirely important, but the key idea here is that you're producing NADPH along the way. Now, the non-oxidative phase starts with this molecule called ribulose 5-phosphate. And it's really not important to know, except for the fact that it kind of sounds like ribose 5-phosphate, right? Which I mentioned before was one of the main primary products of the pentose phosphate pathway. And indeed, it's a precursor for the ribose 5-phosphate. So let's see how that happens. Let's go ahead and scroll down here. So ribulose 5-phosphate is actually broken down by an enzyme, a isomerase. So it's essentially switching around the molecule. It's not really changing the chemical formula, but it's switching around the structure to ribose 5-phosphate. So that's key. So remember, that's one of our main products of the pentose phosphate pathway. Now, another key point of the non-oxidative phase, so we produce, of course, ribose 5-phosphate. Another key point here is that we're also able to interconvert various sugars. So interconvert sugars. And why is this important? This turns out to be really handy for the cell because notice here that there are some products like fructose 6-phosphate and glyceraldehyde 3-phosphate and fructose 6-phosphate that you might be familiar with that come from glycolysis. And remember that these are not all 5-carbon sugars, right? You know, glyceraldehyde 3-phosphate is actually a 3-carbon sugar. So the ability to interconvert sugars through enzymes like the transaldolase and the transkeolase will essentially allow cell to produce more ribose 5-phosphate for DNA and RNA synthesis if needed. And now I do want to say this with one caveat, which is although these glycolytic intermediates can be reinterconverted into ribose 5-phosphate, they cannot go all the way up the pathway to glucose 6-phosphate. So these oxidative phase reactions are irreversible, so shown by kind of the unidirectional arrow, but the non-oxidative phase, of course, allows interconversion and hence is kind of thought of as more of a reversible pathway. So that in a nutshell is the pentose phosphate pathway, and I'll return to the kind of main slide at the beginning and just remind you that the key takeaway is that we are producing a pentose sugar, ribose, and a phosphorylated molecule, NADPH, in this pathway, and that the most unique part of this pathway is that even though we classify it as part of carbohydrate metabolism because it utilizes the metabolites from the breakdown of glucose, there is no ATP consumed or produced in this cycle. So that's what makes the pentose phosphate pathway unique.
Okay. Yep. Wow. There's the CO2 gas. Okay. Cool. Yeah. Let's go ahead and do the reading, and when we come back, we'll start the questions and then exercise. Cool. What's this one? Maybe it is. Maybe it's not. What the heck? Cool. Using the fees code. What? It's strange. I don't know. It should have been late. Or we should have had. Because,
It's kind of strange that we skip from that to this. But okay. Conduction.
Hi, it's Paul Anderson, and in this video I'm going to talk about the action potential, that characteristic depolarization and repolarization of an excitable cell. I'm going to be talking about neurons, but we could also have action potentials in muscle cells coordinating movement, or in cells of the endocrine system, for example, coordinating the release of insulin. So if we think about a neuron is getting information from its dendrites and cell body. Remember, they're connected to a lot of other neurons, and based on the summation of all of that information in neurotransmitters, it's either going to fire an action potential down the axon or not. And so in this video, we're going to think about three different potentials. The resting potential, that's the resting potential of a neuron at rest. It's not really at rest. We then have a graded potential based on the stimulus that that neuron is going to get. And then we may or may not have an action potential. Now that can lead to synaptic activity on the next neuron. I'm going to go through it in the following order. First, we're going to talk about what a resting potential is and how the sodium-potassium pump and the permeability of different ions in the membrane is.
Yeah, that's it. Where's my pretty good actually? Okay, let's go back. Modules. I want to just read the book. Oh, where is it? I think I read it. Didn't I? Sort of. Actually, maybe I can just read it from some from here and here and I think that might be all. I'll do these on Saturday. I'll just do all of them on Saturday. Let's continue.
Contributing to the resting potential. We'll then skip forward to the action potential and the importance of voltage-gated channels. And then we'll come back and talk about how chemically gated channels contribute to either the firing of an action potential or not. And so what is potential? Potential in physics is simply a separation of charge. And so if we think of a battery, a 9-volt battery having potential, that means that there's a separation of the charge within the battery itself. And it also means that we have potential to do work. And so if we're thinking about a cross-section of a neuron, it also has resting potential. What that means is there's a separation of the charges. Now that separation of the charges is going to be across the membrane, just on the surface of the neuron, but we can measure the voltage. It's negative 70 millivolts in a typical human neuron. And so when you're seeing pictures of an action potential like this, on either side of the action potential, we can see that that neuron is at rest. Now, it doesn't mean that it doesn't require energy to do that. And so let's zoom into the surface of that neuron and see what's going on. Now, I've got a little mnemonic that I use with my students to remember the important ions on either side of that membrane. I say the neuron is like a salty banana. Most people remember that a banana has a high amount of potassium on the inside. Doesn't mean that it doesn't have potassium on the outside. There's just a higher percent of potassium on the inside. There are also going to be negatively charged anions. For example, proteins on the inside of the neuron are going to have a negative charge. And then since it's a salty banana, on the outside, we're going to have salt. We're going to have sodium and chloride ions on the outside as well. They're going to be at a higher percentage. We still have them on the inside, but a higher percentage on the outside. Now, why do we have more sodium on the outside, more potassium on the inside? I'll show you in a little bit how the sodium-potassium pump contributes to that. But as you look at this right now, there's still no potential. We don't see a separation in charges. And to really figure out how that works, let's remove a lot of it and just focus on the potassium itself, because it's really the potassium and the permeability of that potassium that's establishing much of that membrane potential. And so we've got a channel on the membrane surface. It's called a leak channel, and it's a leak channel that allows potassium to move out. And so if we think about where is the potassium going to go, there's a chemical gradient from the inside to the outside. In other words, there's a higher concentration of potassium on the inside than on the outside. And since some of it can leak through, it's going to go in that direction. But watch what happens as the potassium moves out. We all of a sudden see a separation in charge. Since this is a positive charge here, it's moving out. We have a separation of that charge, and it's a relatively negative charge on the inside. We also see the arrival of a new gradient. That's the electrical gradient. Since we've got all these positive charges here, they're going to repel each other. They're going to want to move towards the negative charge. And so as more potassium leaves, you can see that electric charge is increasing. And so eventually what could happen is it could start to flow back in. And so we're going to have movement of potassium inside and outside, but we've got an electrochemical gradient outside. Now, why doesn't all the potassium just flow out? It's because these are leak channels. They don't allow much of that potassium to flow out. Now, if we look at a PHET simulation, and I'll put a link to that in the description down below, they've simulated the passing of potassium in and outside of the cell. You can see that we also have another leak channel right here. It's not as busy, but that is a sodium leak channel as well. And so we have an electrochemical gradient of sodium. Remember, there's more sodium on the outside that's coming in. And so we've got these two gradients that are set up. You can see the chloride's not moving. You can see that the proteins are not moving as well. And that's because we don't have a channel for them. Now, if we were to just let this go, eventually we would lose that potential because all the sodium would come in. And so we have a sodium-potassium pump where we cash in a little bit of ATP, and that's going to move that sodium out, 3 sodium ions for every 2 potassium ions. And so as this is moving and establishing that membrane potential, again, it's the permeability of these 2 ions that's establishing it. That sodium-potassium pump is essentially keeping up with the sodium that's flowing in and the potassium that's flowing out. And so that is our resting potential. Negative 70 millivolts is established because we have a separation of those ions on the inside and the outside, and their different electrochemical gradients are establishing that difference in the charge. And so we have this resting potential. Now, it requires energy to do that. But what could that resting potential eventually lead to? An action potential. Now, let's look at what an action potential looks like. This is that PHET simulation again. And you can see there's a huge amount of motion between the ions on that surface. And as we zoom in, we see our sodium leak channel, we see our potassium leak channel, but we see another couple of channels that are really, really important. And those are the voltage-gated channels. How do they work? Well, let's look at the sodium voltage-gated channel. So it looks like this. It's got a little polypeptide ball on the bottom. And so when the voltage approaches negative 55 millivolts, it will open up. And it opens up wide. And that allows a huge amount of sodium to come into the cell. You can see that rush of sodium in that simulation. Eventually, as we move away from that voltage, it's going to be inactivated, and then it's eventually going to close. There's also potassium voltage-gated channels as well, and they're going to open when the voltage becomes around 30 millivolts. They'll open wide open. What is that going to do? It only allows the passage of potassium. Potassium is going to move outside of the cell. And so as we look at that action potential, once we hit that threshold at negative 55, all of those sodium ion channels, the voltage-gated channels, are going to open wide open. What does that do? It allows a rush of sodium into the cell. What does that do to the voltage? It's going to depolarize that neuron. The voltage is going to increase radically because we have an influx of all of this sodium ions. What eventually happens is we get to 30 millivolts, and those sodium voltage-gated channels are going to become inactivated, and now the potassium channels are going to open wide open. What's going to happen? We have a rush of that potassium outside of the neuron. It takes a while for those to eventually close, and so we then have an undershoot. And what's happened is at this point, the voltage has gotten really, really low, but now that permeability, again, those leak channels are going to reestablish that gradient again to negative 70 millivolts, so we could have another action potential. And so if we look at what that looks like in slow motion, we've got a quick opening of the sodium voltage-gated channels. You can see an influx of sodium from there and adjacent. That's followed quickly by an opening of the potassium channels. We then have that outflow of that potassium, and then you can see that permeability is going to reestablish us at that resting potential. We could look at what this looks like as we graph the potential over time. Open of sodium, we see a rapid depolarization, opening of the potassium, a repolarization, and then we have that undershoot. And during that time, until we reestablish that gradient at resting potential, we can't have another action potential. Now, so far, we've only been looking at one cross-section of the neuron itself. How does that message move all the way down the axon? Well, if we look at a simulation, it looks like this. Let me slow that down. What really is going on is we're having depolarization of a segment of the axon, which triggers depolarization of the next segment and the next segment and the next segment. So let's zoom in and look at those voltage-gated channels and see what's going on. So if we look at, this is near the axon hillock at the beginning of the axon itself. We've got some voltage-gated sodium and potassium channels. And so when we hit that threshold, those sodium channels are going to open wide open. We have an influx of sodium ions here, and what they're going to do is they're going to move away from each other since they are going to repel like charges, and they're going to change the voltage in this next segment. What does that do? It hits threshold again and opens up the next sodium channels. Now, why isn't it going to move to the left? Because to the left, we have that repolarization of that segment to the left. So we're opening up those potassium channels, and we have an outflow of those charges. We're not going to be near that threshold for the sodium voltage-gated channels. And so what we're really having on the leading edge of this is a positive feedback loop. We have more sodium coming in, which triggers more opening of those sodium channels. And behind that, we have the undershoot of as we reestablish that gradient again. And so it's going to travel all the way down the axon. Now, this is a simulation of what that electrochemical signal looks like as it moves down. But we have some really long axons in our body. We have an axon that goes all the way from the base of our spinal cord down to our foot. And so it's not going to travel fast enough. And so in a lot of those neurons, they're myelinated. And what that means is they're wrapped with myelin, which insulates that nerve fiber. And so if we look at it like this, what's really going on is we're putting those voltage-gated channels in between that myelin sheath. And I'm not going to get into the specifics of what's going on in here, but if we look at the simulation right here, what it does is allows that electrochemical gradient. You can compare the two to travel more readily as it moves farther down the neuron. Now, action potentials succumb to what's called the all or none law. And what that means is that an action potential will occur or will not, and it doesn't depend on how much of the stimulus it gets. It depends on if you hit that threshold or not. And so once we hit that threshold, we can move towards it, we can move away from that voltage. But once we hit negative 55 millivolts, we're going to have an action potential, and that action potential is going to look exactly the same. Now, in this diagram right here, we have the three different potentials that I'm talking about in this video. At the beginning, we have the resting potential. Remember, that's contributed to by the permeability of the membrane to different ions. We then have the action potential, that rapid depolarization and repolarization of the neuron. But what's going on in here, we have graded potentials. In other words, the potential is moving towards or away from that threshold. And so if we think about what's going on there, to really understand that, we're getting information from other nerves. There are synapses here, and those synapses are releasing neurotransmitters. Those neurotransmitters are moving across the synapse, and they're opening up a new form of channel that's important when we talk about graded potentials. Those are chemically graded channels. And so these neurotransmitters are going to dock with proteins on the postsynaptic side of those neurons, and what they'll do is they'll open it up. What does that do? Well, it depends on what type of neurotransmitter it is and what type of receptor it is. There are some that are going to be excitatory. And if we look back at our graded potentials, excitatory means that they're moving us towards the threshold, towards that all or none action potential to occur. What's the easiest way to do that? If we open up the channel and we have a channel that allows sodium to run in, that's going to depolarize us and it's going to move us to that threshold, just like those sodium-gated channels are going to do. And we also have inhibitory neurotransmitters and receptors. What are they going to do? They're going to move away from that threshold. What's the easiest way to do that is to have a receptor that's going to allow potassium to rush out, or you could allow chloride to come into that neuron itself, and it's going to push us away from that threshold. So we can think of a neuron as an analog portion and a digital portion. So since it's all or none down here, where you're either going to have the action potential or not, so it's like a 1 or a 0. But if we look on this side of the neuron, where the cell body is and the dendrites are, we're getting a lot of information from neurotransmitters of neurons that are attached to the dendrites and the cell body of this neuron itself. Some of those are excitatory. What they're doing is depolarizing that neuron. They're increasing the voltage, pushing us closer to that threshold. And some of those are going to be inhibitory. They're pushing us away from it. And so it's the summation of all of those signals that's telling that neuron to either fire an action potential if we hit negative 55 or not. And so that is the all or none law. And so in summary, what is the resting potential? It's negative 70 millivolts. It's where a neuron is sitting. It has a potential to do work. It's established by the permeability of the membranes and the sodium-potassium pump. We then moved on to the graded potential, and that's based on all the neurotransmitters that we're getting from connected neurons. And then if we hit that negative 55, we're going to have an opening of those sodium ion channels, and we're going to have an all or none action potential that runs down to the neuron. So that's the action potential, and I hope that was helpful.
That was very helpful. Thank you. Interesting. Oh, you what? Oh, interesting. Yeah. Oh, right. Okay. Oh. Sodium. Yeah. Beaches. Depolarization. Myelin. Okay. Cool. Let me see here. It's gonna be transport, communication, endocrine system, neural conduction. Wow, how many modules is that? It's kind of quiet. That's okay. And then I think I have the web exam one. Are these open, by the way? Oh, no, these are locked. Sure. Where was I? Contents. Yeah, I'll just show you guys this one right now. It's this one, and then I'll do this one, right? Yep. It'll be these two. Oh, let me take a look at the. Let me take a look at the other one. We need to figure out when the other ones are due. Okay, also the 22nd. How many? Oh, just those three. Can I do it? I think it looks like I can do it without it, huh? Because I don't have the. I think so. Alright, well, whatever. Let's do it. Oh, man. Oh, my goodness. just read these. All right. They're pretty short, so it shouldn't take that long, and then we'll get back to the other one. didn't see it from before.
transition state always exist in a higher energy state than the reactants.
Your paper, just get Grammarly. It's free.
I don't need.
Where does the energy for chemical reactions come from? Many chemical reactions, even exergonic reactions, require an input of energy to get started. On ancient Earth, before the formation of macromolecules, chemical reactions were powered by lightning, sunlight, and heat from molten rock. On the modern Earth, living organisms, including humans, most often obtain the energy they need from other molecules. Cells use the energy in these food molecules to power chemical reactions. However, the energy cannot be used directly. It must be transferred first to an energy-carrying intermediary, such as the compound adenosine triphosphate, or ATP. When chemical bonds in ATP are broken, energy is released. This process provides the energy needed for chemical reactions, which power life. This graph shows a hypothetical reaction between atom A and a molecule containing atoms B and C. The activation energy, EA, is the amount of energy needed to get the reactants to the transition state in which bonds are broken and new bonds are formed. This transition state usually lasts for a fraction of a second. In this example, the products of the reaction are the molecule AB and the atom C. Because this is an exergonic reaction, some of the energy stored in the reacting molecules is released. Often, activation energy can be a barrier that prevents a chemical reaction from occurring, except very slowly. The activation energy for many chemical reactions can be reduced with catalysts. Without catalysts, many of the chemical reactions in metabolism would occur too slowly to support life. Catalysts stabilize the reactants in the transition state, allowing old bonds to break and new bonds to form. Even though the activation energy is lowered by a catalyst, the amount of energy released by the reaction is unchanged. In living organisms, most chemical reactions are catalyzed by proteins called enzymes. The three-dimensional structure of an enzyme is crucial to its function. If this structure is disrupted by exposure to high temperatures, for example, the enzyme stops working. The enzyme binds reactants in its active site, which is specially shaped to fit the reactants. The reactants on which enzymes act are called substrates. In this example, glucose is the substrate of the enzyme hexokinase. The enzyme and substrates are represented here in an artist's drawing. The substrates include the atom A and the molecule BC. The substrates enter the active site of the enzyme. By holding these atoms close together, the enzyme reduces the amount of energy the atoms need to enter the transition state. In the transition state, the bond between atoms B and C is broken, and a new bond between A and B is created. When the new bonds form, the products are released, and the enzyme molecule is ready to catalyze another reaction. Each enzyme is specific for only one reaction. For example, this enzyme could not catalyze a reaction between atom A and molecule BD. The fit between an enzyme and its substrate is described by the lock and key model. The enzyme is the lock, and the substrate is the key. Only a specific substrate will fit the enzyme's active site.
Yeah, that makes sense. Okay. Yep. Wow. Okay. Maybe we can read that like 7:45 or something. I just want to go through these. I didn't really get a chance to be honest. So might as well now we're here. Plus, it's just a different take on things, you know. I mean, the more, the better. It's a bit too much, eh? How about this one? This is too much time. Just Yeah. Yeah. All right, let's take like 5 minutes and then we'll read this one. Yeah, and after we read this one, we'll go back. We'll go back and we'll finish the other two. I guess I can just put this here. Yep. All right, we'll take a 5 minute. Hey, Ricardo. It's first week of August. Thanks, Sila. Appreciate it. Cool. Yep. Yeah, all right, let me 5 minutes. And then we'll finish this one. 1277. I think at least by now to read all this. It's okay. We'll do it. Gotta do it. Knowledge. Yeah. But that's okay. Sorry guys, I had to feed my dog and have to do some other stuff. We got some milk. Milk. Yeah. Nope. Yep. That's good stuff. My bad, guys.
It's got something here.
Vitamin B3. into. Yeah, it's okay. We'll keep reading this one. I like this one because it has more connections, and I think the other one, I mean, with Khan Academy, well, both are good. I mean, I'm just gonna do both. That's fine. I'm just talking. I'm just talking. Yo.
is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
North Dakota State University. Cool. hydroxyethanol group. Yeah. Pigeon flight muscle. What the heck? Wow.
on your own time.
Each loaded into the mitochondrial matrix acetyl group from acetyl CoA2 NADHs, those produced by
I
glycolysis.
can't tell. I don't remember if we watched this one or not.
Important process.
I don't think so.
To produce.
Did we?
Aerobic respiration is the most important process used to produce energy for a cell. It involves several pathways, including glycolysis, pyruvate oxidation, the citric acid cycle, and electron transport. Various sugars, fats, and proteins enter these pathways, and they can all be broken down to produce ATP energy for a cell. In this animation, we will focus on the citric acid cycle, also called the tricarboxylic acid, TCA cycle, or Krebs cycle. The citric acid cycle takes place in the matrix, or fluid, of the mitochondrion. This is where mitochondrial DNA is found, and where fatty acid breakdown takes place. The citric acid cycle involves 8 chemical reactions that use acetyl-CoA and oxaloacetate to produce carbon dioxide, NADH, ATP, and FADH2. The NADH and FADH2 are electron carriers that can be used in the electron transport chain to make more ATP for a cell. Let's take a look at the major steps of the citric acid cycle. In the first step of the cycle, a 2-carbon molecule and a 4-carbon molecule are combined to form a 6-carbon molecule. That 6-carbon molecule undergoes multiple biochemical changes during the cycle, and at the end, the original 4-carbon molecule is produced. Each time a carbon molecule loses 1 carbon, a carbon dioxide is released. So 2 carbon dioxides are formed during the conversion of the 6-carbon molecule back to the 4-carbon molecule. So where does the 2-carbon molecule come from to begin the citric acid cycle? This molecule, acetyl-CoA, is made from pyruvate. Pyruvate is a product of glycolysis, the other major ATP-producing cycle in the cell. Pyruvate is transported into the mitochondrial matrix, where it is oxidized to acetyl-CoA by pyruvate dehydrogenase. This step produces 1 NADH and 1 carbon dioxide for each of the 2 pyruvate molecules made from glucose. The acetyl-CoAs are the starting point for the citric acid cycle. In the citric acid cycle, the acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. 4 different enzymatic reactions then lead to the formation of succinate. These steps produce 2 carbon dioxides, 2 NADHs, and 1 ATP. Succinate is then recycled back to oxaloacetate through 3 more reactions. These steps produce FADH2 and 1 more NADH. All of the energy molecules made in the citric acid cycle, along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
Wait, what?
As molecules made in the citric acid cycle, along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
Thank you. Alright, 5 minutes on the clock. can't enumerate battles won and battles lost. I can't enumerate the cost. Let's take a break and exercise. This is gonna get way too hot though. Hold on.
Just want to think of my own thoughts. That's good.
Alright, 50, 65, 65, 66, 33. All right, let's do max push-ups in a minute. Right. All right. I'm gonna take a quick shower. I'll be back. Excuse me. Pretty cool stuff, huh? All right, I'll be back. Take a quick shower. Cool. Yep.
Hey, what's that sound? Everybody look what's going down. We little lion man. Someone that'll never be what is in your heart. Weak little lion man, you're not as brave as you were at the start. Break yourself, take it.
All right, let's at least finish it up here, eh? Yep. Yeah, that's good. Can I use the same ones? I think I can. Yep. Ah, that's good. Okay. Excuse me. So sorry. Did you read this already? We read this in Khan Academy. It's funny. Okay. That's actually quite funny because this is almost the same thing they have. All right. Yeah. I think we're going to stop here. I'll pick it up tomorrow morning. Yeah, we almost got through it. It's all right. We can wrap it up tomorrow and start the rest. All right, everyone. Thanks for watching. Peace.